Good evening, everybody, and welcome to ROV SuBastian, Divestream 913. We're going down into the midwater again, initial depth of 700 meters, and once again, we'll be imaging, scanning, and sampling various midwater organisms, also looking for eDNA genetic information. Anyway, enjoy the dive. I'll pass you over to Kakani Khatija, one of the engineers from Mbari, who is currently leading the dive. Okay, Kakani, you're on.
Heard you. Thank you, Jimbo, for the lovely intro and for getting us live. Excited to be here. I am not in this chair very often. I'm not sure why. But feel free to ask questions. Just a friendly reminder that I am an engineer. I'm a wannabe biologist, so be careful with your questions because there's only so many things I can answer, but I can route them to other people on this boat. We're joined by Joel and Marco, who are our pilots. And we've got, that's really the important people in this room.
Forgot the name of that other guy.
But we are off the coast of Brazil still. So I think there's a command. There you go, the vessel tracker.
Translate to Spanish?
Yeah.
Anytime if you hit ! location in the chat, the Nightbot will respond with a link which will give you the vessel's location.
Para nuestra invasión número 913. Quien nos habla ahora es Julieta, de la Furgi. Estamos acá en la sala de comando. Quien está comandando la inmersión es Kakani. Nuestros pilotos son Marco y Joel. Jimbo está en la transmisión. Tenemos acá con nosotros al profesor Renato Nagata, también de la Furgi. Cualquier duda que tengan, o pueden dejarla en el chat. Estamos acá para intentar responder lo máximo que podemos. Obviamente el barco cuenta con un montón de especialistas en las diferentes áreas, como habrán podido ver a lo largo de hoy y de ayer. Tuvimos varias colectas súper copadas. Ayer a la noche incluso colectamos un cefalópodo, fue una locura. Todo el mundo súper emocionado y varias otras especies de individuos que creemos que pueden ser especies nuevas. Acá Joel les manda saludos a todos.
Saludos a Pablo Daniel 755.
Estamos ahora yendo hasta los 700 metros de profundidad y luego ahí vamos a empezar nuestra búsqueda. Obviamente les recordamos que estamos en aguas internacionales, fuera de la zona económica exclusiva de Brasil. A la altura de ahora creo que estamos un poco más al norte de Alagoas. explorando los organismos del mesozooplancton, que va desde los 200 hasta los 1200 metros de profundidad. Antes de lanzar de la inmersión del ROV, tuvimos una colecta de red de plancton.
Do you know what language people are asking that question in? I feel like that symbol just indicates it's a different language, but I just don't know which one.
I don't know how to find out.
Oh, okay. Well, there's a question in the
Yeah.
chat.
But at some times they can all translate to English, but I don't know how. A couple of days ago they came in
Spanish? Or
or
Portuguese.
Spanish or Portuguese.
Well, maybe we have it, yeah, because I'm not sure. So there's a question in the chat about why did we pick this time to go deep, or this time for the dive? It is dark in Argentina. It's also very dark here. One of the reasons why we're trying to do these dives at night is because we're trying to capture the animals that are involved in these mass migrations at night, known as diel vertical migration. But we also have the luxury of being able to dive all day, every day, while we're here at sea, thanks to having 24-hour operations of the vehicle. So we're trying to get as much time in as possible while sampling these midwater communities.
What you can do is bring up the dive on YouTube, pause the video, and chat it to the live.
Well, does it do the translation?
Oh, it shows you the raw language, is what I'm saying.
The original.
Yeah.
Oh.
Sorry, Juliet.
Hi, how are you?
But if you bring it up on your computer or laptop,
We are going.
Yeah.
and then just pause the video, then it's not using tons of bandwidth, and you'll see the chat.
Okay.
And then be able to tell what language it's in. Hopefully.
Yeah, most of them are in Spanish.
Okay, rather than Portuguese.
Ahora nos encontramos bajando hasta los 700 metros de profundidad y una vez que lleguemos ahí vamos a empezar nuestra búsqueda. Acá siendo las ya 8 de la noche, estamos todos bastante perdidos con el horario, está súper oscuro obviamente. Acá a esta altura amanece a las 5 de la mañana, ya está súper claro, y a las 5 y poco de la tarde ya es prácticamente de noche. Quien quiera saber más sobre sobre nidarios, sobre medusas, quien está acá con nosotros es el profesor Renato Nagata de la FURGI, especialista en estos organismos, que ha estado juntando un montón de cosas en los últimos días y prácticamente ya no duerme.
Olá a todos, professor Renato Nagata, da Universidade Federal do Rio Grande. Aqui é um deleite para os que estudam as medusas, muitas espécies. E agora nós vamos até 700 metros de profundidade. É um momento em que o zooplâncton já migrou para a superfície, porque à noite eles vão à superfície aproveitando a escuridão da noite para se alimentar. Espero que nossos colegas que falam espanhol possam entender o meu português também. Vou tentar falar claro e devagar, despacio.
I'm here now. I've been changing.
Portuguese mic.
Japanese mic. American English.
Bueno,
Chloe
sobre
san.
la pregunta de
Nihongo
Pablo
de.
Daniel 75
Nihongo de.
sobre los indicadores de microplásticos en estas campañas, sí aparecieron algunos en las redes, tanto en la red de Tucker como en la de Plankton, aparecieron unos pequeños, obviamente microplásticos de algunos colores diferentes. Yo los estoy separando, que trabajé durante mi doctorado con eso. No han aparecido tantos como por ahí esperaba o como aparecen en otras regiones. También tenemos que considerar que estamos prácticamente en el medio del océano y súper alejados de la costa.
What's the closest city in Brazil to where we are?
I think it's close to Alagoas.
I think that we are upper Alagoas.
Up, yeah, we passed through Alagoas already. Yeah. Maybe João Pedro. Let's check.
Estábamos debatiendo a ver a qué altura estamos. Me parece que ya pasamos a Alagoas, en el nordeste de Brasil. El crucero termina más o menos a la altura de Río Grande do Norte, por ahí. Nos quedan algunos días de trabajo y después, una vez que el barco termina, seguimos directo para Fortaleza, en el estado de Ceará, en el nordeste de Brasil. Sí, las corrientes los transportan, obviamente, también en relación a los microplásticos y la pregunta de Pablo. Los transportan. Hoy en día estos tipos de residuos están en todos lados. Incluso encontramos, no tiene nada que ver con el Falkor 2 y la expedición, pero también encontramos microplásticos en Antártida, en focas, que era lo que yo hacía durante mi doctorado. Así que obviamente podemos decir que
That was really interesting that we came across a big
están en todos lados, infelizmente.
We might want to see. Ooh,
Was that big house, or it was very round?
it looked gelatinousy to me. What did you see? Oh my goodness.
Need him somebody's jelly.
Was it a jelly?
I'm a computer dude. What is it?
Come on, I'm an engineer.
Computer dude Jimbo. Look jelly.
You can replace all of that.
It was globular.
Yeah.
Okay,
Yeah, where is
we
the
are
Kraken? I would love to know.
we
and
are
you'll be able
close
to find it.
João Pessoa city, which is the easternmost city.
Yeah, we've got the first, 9 dives in now?
I think.
Yeah, we're only 1 dive behind it.
Yeah, 9 dives of data is already in Fathomverse. I suggest getting on soon, because it's such a treat. We don't actually have a lot of midwater data. And if you have no idea what we're talking about, Fathomverse is an app that allows you to take this journey with us, help us identify all life in the ocean, and train better algorithms so that we can accelerate processing of all this visual data, like the dive that you're watching right now.
Traduzindo para o português, Fathomverse é um jogo desenvolvido pelo pessoal do Monterey Bay Aquarium, no qual você ajuda o pessoal do Monterey Bay Aquarium Research Institute a desvendar a biodiversidade dos oceanos profundos. Você baixa esse aplicativo, pode ser no Play Store, na Apple Store, e você se loga e joga, né, catalogando, ajudando a catalogar a vida marinha, né? Você faz um treinamento, aprende a identificar animais marinhos, né, animais marinhos que aparecem, E aos poucos você vai ajudando nessa catalogação de animais que aparecem nos mergulhos científicos realizados pelo pessoal do Monterey Bay Aquarium.
Respondiendo a la pregunta de Pablo y pasando para el asunto de los microplásticos de vuelta, sí, los microplásticos ya alcanzaron todos los substratos posibles. Están en el aire, están en el sedimento, están en la columna de agua, están en la superficie, están en las playas, en los organismos y en el hielo marino. Y como Renato también estaba traduciendo lo que Kakani contaba, Fathomverse es una aplicación que fue desarrollada por el Instituto
Monterey Bay Aquarium Institute.
Gracias, Renato.
Research Institute.
Que les permite obviamente descargarlo en el celular y es una especie de algoritmo en el que los jugadores de esta aplicación lo entrenan para identificar diferentes especies y facilita en el día de mañana a los bancos de datos de las especies de la biodiversidad marina.
Do you like pulse or illuminate more? Neither.
Identify.
I'm an identify person. And I know. There's one way to figure that out, Jimbo. It's to play the game, but I do not want to ruin it. I think what's really great about the Fathomverse app is, everybody is looking at, either mostly imagery that a lot of people haven't looked at, a lot of animals that aren't identified, and we together have the opportunity to identify them. And within Fathomverse, what we've done is provided multiple ways for you to experience or view that data. And we've done that through creation of different, what we call mini games. And so Pulse was the very first mini game. And this is a mini game where you're basically, think of yourself like a vehicle underwater, swimming around in currents. And as you're coming up across animals, you can tap your screen to open up those images. And there you get to see, things sometimes for the first time. And then you collect animals and then later ID them. Illuminate is another visualization, again, very similar to this experience, like what you're seeing today. And the idea that we're essentially underwater in the deep ocean with a vehicle that is equipped with what's essentially a flashlight, and we're just bumping around in the dark. And so that's essentially what Illuminate is, where you as a player are illuminating different parts of the screen, finding animals, opening up images, and then collecting them. And then there's two more mini games. One is called Identify, which kind of gets rid of all of that, fun, gamey stuff in terms of going out and searching for different types of animals or images, and just sends you all of the images at once. So that's a team of 4. And can you go to the right? I think I saw something pretty big, but we might have gone right past it. And when you get to Identified, you're just shown all of the images in that set, and then you're asked to classify them. And so those 3 games involve classification. So like identifying, basically putting a label or naming an image. And I think we have, 50 plus types of animal groups that you're able to assign names to. So that's those. And then last year we introduced a new mini game called Bound. And Bound is really important because it allows us to look at a whole image, like the scene that you see here, and in a whole image be able to locate different animals or targets of interest and indicate where in the image those things are. So what that data type is called localization, or drawing essentially a bounding box around those targets. And it's these two data types, localization and classification, that's needed for us to train machine learning algorithms that will allow us to very, very quickly extract useful information from video like what you're seeing now and quickly label it or name animals in it or count those animals. With the goal of us one day being able to do that kind of work really quickly anywhere in the world's ocean. And so we're still a long ways away from, I think, realizing that goal, but already within the past year or two, we've seen, thanks to contributions of, that's a jelly. Can we zoom on that, please? I think we lost that other one. We've already been able to improve the models that we use. We've already made contributions to global image repositories like FathomNet.
Agora uma água-viva muito bonita, com aquela típica coloração de animal de água profunda, né? Vermelha. É provavelmente uma água-viva coronate. Agora uma visualização melhor vai nos permitir ter uma melhor identificação desse animal que está vivo, pulsando aqui na nossa frente. Olha só.
Any guesses in the chat what we're looking at?
Tenemos una
Tolidae.
agua-viva
Parece ser
en
perifila.
pantalla, una medusa.
We could try.
Estamos intentando mejores imágenes para ajustar la identificación con un color bien típico
I know, right?
de animales
No
de agua
pressure.
profunda.
Não vou me arriscar, parece perifila, mas tá
Paraphyla is the
tá um
guess.
pouco diferente.
I think that's a good one. I think, yeah, we're going to want to do an iris. Yeah.
Kind of were settings loaded.
Yes.
Certamente
So,
uma água-viva da
We're
família
currently trying to
Perifilidae.
get the animal to that instrument that you see all the way to the left. That is IRIS, EYERIS, where RIS stands for Remote Imaging System. And this is a light field camera or planoptic camera, which gives us three-dimensional data in a single frame. Yeah, you can have whatever lights you want.
The corner is kind of dark.
This animal's pretty small, so we'll see what it looks like.
That's not a problem. I can zoom in.
Tuvimos la
Yeah.
explicación de Kakani,
Don't do
que
that to
obviamente es
Hillel.
una de las que desarrolló
So paraphyla or
este
paraphilopsis.
equipamiento IRIS que toma
Pode ser.
varias imágenes al mismo tiempo y las acopla en solo una base de datos, y lo que permite generar imágenes 3D.
So he's right
Cualquier
now
duda del
nice.
iris,
So he's a little bit close.
tenemos
That's good.
a la
So green is
creadora
the
experta
sweet spot.
acá en la sala de control.
Yeah. Might want
All right, I
to
changed the range.
zero the what? What do you change it to?
Parece que es bastante chiquita. Ahora están intentando tomar
So maybe
las
50 to
imágenes.
300 was right.
Could be. And if so, I will say
Hay
yes, you were right.
What?
And if so, I will say you're right.
Reserio
No.
también dando vuelta por ahí.
No, let's see if it, because we were resolving blue on the particles, but not on the gel.
Yeah. All right, Joel is lining it up.
La pregunta de Pablo es para Renato.
Now, what you
Estas
guys can't
especies
see is
no
on top
son
of
de
this
estas
camera,
profundidades, no creo que
we
lleguen a la costa.
added
Renato,
another camera that gives a
Não
slightly
chegam.
wider field of
Eventualmente
view.
essas espécies podem morrer e ficar à superfície, flutuando próximo à superfície, às vezes ocorre isso, mas são espécies de altas profundidades.
Como puede pasar,
Paging Dougal.
voy a
Dougal, do you want
sacar
this?
la carta de los mamíferos, como puede pasar en algunas especies de
I don't think
mamíferos
we need
que
the
vemos
PID on this.
encallados en las
I think the
playas.
question
Obviamente
of
no
us
ocurren en
getting,
las
maybe
áreas
some
costeras,
nicer
pero
4K
pueden morir en alta mar y
zooms.
llegar
We've
por
gotten
las
a few,
corrientes
but we might want a
hasta
few
la
more.
costa.
Look, see,
De la misma
it's just
forma puede
magically
pasar con
now
estas medusas.
in iris. You don't even have to try anymore.
All right, how do I change calibration? Calibration.
Stop trying.
No. After you get it.
Okay, so we go for some.
Esse é
Yeah, if we can get some nice 4K
uma espécie
zoom shots.
da família Periphylidae, porém é difícil analisar o animal, é uma espécie pequeno, não dá para dizer se é do gênero Periphylla, que é mais comum, mais conhecida, água-viva de águas profundas com essa coloração, ou Periphylopsis, que é um outro gênero da mesma família.
Paraphilina? Oh yeah, it's really close. Really close.
Periphylla.
Doesn't look like.
Certamente é um indivíduo juvenil.
I've pulled up the deep sea guide, and of
Uma
course
coisa
it's
muito
data from Monterey
interessante
Bay, so it's not
e parece ser
necessarily
perifila mesmo.
transferable to this, but
Uma coisa muito interessante desses animais de águas profundas é que eles têm um ciclo de vida longo.
Aeginura grimaldi.
Como?
Aeginura grimaldi.
wow,
Oh, Dougal is here.
Dougal's here.
Dougal.
We need it. We don't have one yet.
We need it.
All right. Desampling.
Do we need to do a?
It
maybe we
normally
can-
sits like this with the
Yeah.
tentacles completely out horizontal when it's feeding.
Yeah.
If it takes that position again, then that's a cool thing to have.
For the-
Otherwise, it's just gonna keep swimming with the tentacles below it.
Okay.
That's
Do
a cool
we?
thing to have how.
Is it possible to test for bioluminescence?
Yes.
If we turn.
If you collect it,
Just do that in the
if you
lab.
collect it,
Yeah.
No, that we will do anyway
well
later.
then great.
I meant
I just don't want us
native.
to. Yeah. I
But
don't
if
know.
you guys feel that you will lose it
That
by
would be my
switching the light
concern.
off, then don't do it.
Oh, yeah.
That would be my
Yeah, because the
concern.
flashes.
We'll do it on a big one when we get one.
Yeah.
Yeah, the flashes
Yeah,
are
should we
really
just try
bright.
a PIV scan then? Because I wasn't sure what we wanted to do with this.
The top of the bell is kind of transparent.
Yes.
Ok,
And then it becomes pretty
corrigindo
dark.
aqui a
We'll give it a
identificação,
try. You ready?
professor Dougal Lindsay nos comenta que, na verdade, trata-se de uma hidromedusa de Aeginura grimaldi. É uma narcomedusa, uma água-viva típica de águas profundas que tem tentáculos apontados para cima, né? Então é um animal
Aeginura?
que é
Aeginura?
muito pouco conhecido também e vamos tentar fazer a coleta.
Cromaldi?
Aeginura grimaldi.
Okay.
Como
I just
Renato
remembered,
dijo,
it's the weirdest thing.
y traduciendo un poco, tenemos acá una hidromedusa.
We have a confirmed ID on this animal. It's Aeginura cromaldi. Yeah, me too, Lisa, me too.
Un animal poco conocido de aguas profundas,
Oh, that's bright.
que tiene los
Grimaldi.
tentáculos orientados hacia
Oh,
arriba.
yeah, look at
Entonces
that.
vamos a proceder a realizar las colectas.
It's a narcomedusa.
Just gonna go ahead and zoom in. It's not that large.
Yeah, all right.
I'm gonna keep zooming in until it looks big.
Yeah, and we're really not certain if the scan's going to work well on this animal because you can see at the base of its bell, there's a lot of red pigment there. And sometimes when our laser hits that pigmentation, it'll either cast a shadow and prevent light from going through. So we're watching a real life experiment.
Lo que está explicando Kakani es que como esta medusa tiene bastantes partes rojas, puede ser que cree un reflexo en el láser y aparezca la imagen
There's a little light.
con algunas sombras.
Oh,
Oh,
Still in a good area.
ainda não é o momento de fazer coleta.
This is only 3 times zoom. We can go up to 10.
No, thank you.
Oh.
It's good.
It needs to stop for a second.
Yeah, let it go all the way through, otherwise I can't use it.
Going up, it's going up.
I think it's working. I think it's working. You're not seeing a big shadow, are you?
No, it's bright, but not a big shadow. Not sure how much detail is really on the, like, right on the lower bell margin.
Well, especially since they're swimming.
Bit orientation dependent too. Ooh la la.
Ooh, what is that? Oh,
Oh, yeah. This is probably the jelly that looks the most like Blobbert of all the jellies.
Say Blobbert?
Blobbert.
Blobbert.
I think we're all familiar with this character.
Blobbert is a children's book written by 3 people, including Tommy Knowles, who's a curator
An aquarist.
at the Monterey Bay Aquarium. It's about a jellyfish going on an adventure.
Hmm.
Uh-oh, where did it go?
Is there a translation for other
Oh,
languages?
nice.
You know, I'm not sure. We should ask him.
Yeah. Maybe.
It's in English. It has
Can
a
be
few
in-
extra fun bonus facts for slightly
Uh-huh.
older kids who want to learn more about the ocean. But I imagine a translation wouldn't be that hard to do for them.
Cool.
What's the title of the book, yes?
Just Blobbert, Blobbert.
There isn't like a
Blobbert.
subtitle?
Blobbert. He's
Mm.
a little blobby jellyfish. There's kind of fun inside information from Tommy, who had like
Oh,
an
it's
internal
low, it's way
struggle
low.
making this very cartoonish jellyfish
There.
because he looks for all these very accurate details on jellyfish in his
Oop.
professional life, but they decided that as a story it was way more fun to go the more
tenemos
cartoon route.
un pequeño
That is
ciclotónido,
really
me parece que en
Oh, it's
pantalla.
high. I think it went really high. There it is. All right.
En la sala de comando no solo están los pilotos y los encargados del
One
sistema,
more.
está
Jamie says the slices look
también
nice, so I guess we're gonna
Bocacani,
set
que
to
está
right.
comandando la inmersión y decidiendo en qué organismos
It's a
paramos
little
y
low.
cuáles
It's a little low. Let's do that
muestreamos.
again. Oh, it's way inside now.
Y
It's way
tanto
inside.
Oh, getting a little close.
el resto del personal que está acá están encargados de manejar el láser y tomar las imágenes. Y después hay otros
Blobbert's
2
Ocean
investigadores que
Adventure.
están
See, there's more to that.
están
Title.
llevando un control de las especies que vemos en
Oh,
pantalla
that was fast. We got it.
para saber qué
Yeah.
se registró y qué no.
At some point we gotta call it, right?
Yeah, one. Let's collect that animal, please.
Ok, foi feito então o
Big sample,
escaneamento.
This one could probably be fine in a section sampler. Yeah,
O escaneamento do animal
but
através
we'll want to
do
be pretty light
iris.
on the section.
This was DPIV.
Oh, desculpa.
Está pasando
Right,
otra
so what
medusa
you-
también.
Outra medusa, parece ser uma traquimedusa opalonematite.
What you just saw with the laser, or that really bright red light, that is the instrument called DPIV. What we're doing with that is we're illuminating very, very, very thin slices in the water. And what we saw was as the animal's body went through the laser, we were collecting the imagery of that slice. And those data are used to do three-dimensional reconstructions of their bodies or structures. And so if you want to check out some of the models we've already created, nice job. Nice
Suction
job. We've got a
sampler.
collection. Fingers crossed.
Okay.
Robert's Ocean Adventure.
Kakani,
Huh?
how thin are the slices that the
Just
DPIV?
under a millimeter.
Sorry?
Just under a millimeter.
Under a millimeter. Ok,
Yeah, about 0.8.
thank you. Então, a professora Kakani Katija, do Monterey Bay Aquarium, está explicando que
Not perfectly aligned.
esse escaneamento do DPIV consegue realizar fotos ao longo de um eixo e fazer fotos ao longo de várias profundidades de campo de visão, que são menos de 1 milímetro, né, a distância entre cada foto.
So we'll wait for it
Então
to
temos a
settle.
confirmação aqui,
And
tá
check
coleta.
the index now, please.
Pudimos colectarlas.
Nice. Number one was
Primeiro
Midwater.
animal coletado.
Número 1 adentro. Ahora seguimos
Grimaldi.
buscando más.
Go team. What are we going to find next?
Large Salvation House, of course. What's next?
We have enough larvations.
We don't
We
have
came up
enough Fiddleria House.
687, is that right?
We don't have to
You know,
collect it, but we want
That's,
to
I mean,
see
that's fine. I feel like between 600 and 700 meters for a while. So like if we come up a little bit, it's okay. We just haven't been able to spend a lot of time at that.
Sí, la mayoría de los investigadores que están acá son taxónomos, los que trabajan con biología, y después, bueno, Kakani y Just. Are you engineer también? Also?
Oh,
Son
heating nav.
ingenieros. Son los que desarrollaron el DPIV y el IRIS.
E respondendo a Pablo Daniel, sim, a fragata portuguesa chega hasta la costa, mas a fragata portuguesa é um animal de superfície, né? Então, quando temos vento soprando de oceano para a costa, as fragatas chegam à costa e, apesar de que a maioria da população vive em em oceano aberto, né? Então, apenas em algumas ocasiões que as fragatas portuguesas chegam à costa e ocorrem as arribaciones, né? Os grandes encalhes, né?
Okay. So small. Did you look for Dougal? Did you look for Dougal, or did he just emerge?
I went to
Okay.
Dougal is looking at the jellyfish they
You're
collect.
trying to figure out if you were calling the Google signal worked. It did not.
It did not
We
work.
have audio in the wet lab.
Oh.
Vendría a ser más como una tomografía de el escáner que le hacen a los organismos.
Another Keating nav.
Justo en las redes, en una de las redes de plancton me parece que vino una pequeña fragata portuguesa. No sé dónde quedó, pero bueno, la idea también, nosotros, tanto
Keating nav?
Silvina
Another.
como yo, juntamos muestras después de que todo el mundo ya hace sus experimentos para separar algunos pedacitos de estas muestras para isótopos estables, que lo que nos permite es poder reconstruir de alguna forma la cadena trófica de esta región. también con el ROV tiene acopladas unas botellas Niskin que permiten la toma de agua a diferentes profundidades, comandadas desde acá, desde el barco. Y una vez que llegan a la superficie, las filtramos para poder concentrar todo el fitoplancton que hay en la columna de agua y también poder tener la base de esta cadena trófica, los valores isotópicos de la base de la cadena trófica.
Oh,
What is that?
Oh,
Hold on.
larváceo.
Ooh, oh, that is a larvation.
Opa.
This one is big one.
That's okay.
É,
That
muito legal.
that was really surprising to me. All right.
That, but just the house.
Yeah, it's okay. We'll keep an eye out for them again.
Ok,
That looked like a gelatinous thing. It didn't look like a.
acabamos de ver agora
Infrastructure.
um larváceo dentro da sua casa,
Seems like than we had during the day.
batendo a sua cauda.
So it's better or worse? It's worse now, I think.
E é uma casa que ele produz com um muco que ele secreta, né? Um animal que produz o muco através do
monitor
endóstilo.
looks like
E esse muco se transforma
somebody
numa
ran
casa
their head into
estruturada.
it. Yeah.
Just mostly the edges seem to turn kind of
E essa casa estruturada serve para filtração de partículas. E ele bate a cauda, transporta a água e filtra partículas.
close down aperture. So Kakani is happy.
I think the part of the challenge is you can't change the gain on this camera. Maybe if we have.
That's the minimum.
Which are the target species for today? You wanted to collect larvations?
Yeah, well, mostly for scanning is the layer larvations. Rust might still.
Oh, that's a little bit better, yeah.
Yeah.
Always Tomopteris.
But it's
and
so
polychaetes.
grainy.
Yeah, Karen's certainly still interested in those.
Okay.
Let's see what else we can find. Opportunistic.
Qué mamas. Hoy estamos buscando prácticamente larváceos, que son esos organismos que vimos hace un ratito en pantalla y que construyen su casa a partir de un mucus que lo usan para filtrar partículas y alimentarse.
Oh, right.
Algunos las palabras
There's
en
something
español
straight
son
ahead.
difíciles
I
de
think that's a
traducir
little jelly.
cuando estás sobreviviendo en inglés. Son temópteros
Or-
y algunos ketognatos, que son también conocidos como gusanos marinos o algo así. Son
That was a
especies
larvation.
estrictamente carnívoras.
That one was
Los
pretty
temópteros vendrían en pariente de los
Right
moluscos,
now,
como son los caracoles, por ejemplo. Y bueno, todas las especies que parezcan diferentes vamos a hacer lo posible para colectarlas y al menos también escanearlas.
it looks like your shutter speed or exposure, so it's super long.
Ya
I
tenemos
would
bastantes
increase your shutter speed.
o varias especies que creemos que es la primera vez que se registran
Why is it
o que nunca no han sido descritas hasta ahora.
the tracks, like the tails on all the particles? Why is that so bad?
Um grande destaque que está nos surpreendendo aqui neste cruzeiro são as espécies de sifonóforos que estão aparecendo, né? Conversando com o professor Dougal, que é especialista no
Oh,
grupo,
okay. Okay.
tem relatado que as espécies de sifonóforos Fisonec são muito abundantes aqui e diversas, né? Muitas espécies que ele nunca viu, que nós nunca vimos e que podem realmente ser animais novos, desconhecidos da ciência.
Someone is
oh.
happy.
You
Some
want to reduce the gain.
narco.
Gain, you want to reduce.
It's on jelly therapy.
Shutter.
On
We're
jelly
seeing lots
therapy.
of jellies.
Fine on the shutter.
Yeah.
You might want to up your iris.
She's like an addict.
You have to
Yeah, so you have more light now.
It's done. It's completely unlike any other one.
So they have to
Yeah, you have
reduce
to
your
redefine the genus.
error,
split it.
increase your
But
gain.
it sounds like a lot of work.
It's not mature.
Okay.
It doesn't have any gonads.
That's a little bit better.
So, and that's one of the
But,
main
you know, you
things
see how
use to
you
have to
can-
describe it.
Yeah,
So,
no,
it looks
You can
big
control
and
your
bigger.
gain.
Sí, lo que estamos viendo en pantalla, Pablo, es lo que se llama nieve marina,
Which
que son
I'm adding it to my feedback.
restos de
Are you
detrito,
talking about-
partículas, animales muertos que
Let's keep going.
se acaban hundiendo en la columna de agua y sirven de alimento para los organismos que están en profundidades y que no tienen acceso a la luz solar, como por ejemplo el fitoplancton que está en la superficie para hacer fotosíntesis.
That is
Entonces,
the
todos los
siphonophore.
organismos que están en las profundidades
Oh, yeah.
se alimentan de esta y que son filtradores,
Really gallant. There's
se
something
alimentan de esta
big
nieve
on the
marina.
sit cam. So we're going down.
So yeah, we need
Ignore that
mature.
fish.
Hola Dora,
Mature
fiel
to believe.
seguidora de todos los streaming desde que estamos acá,
that.
Oh yes.
Keep going.
Oh.
Sorry.
que estamos viendo acá en pantalla.
Look at
Oh,
that.
siphonophore.
The Bergmania?
Siphonophore. Yeah, can
Otro
we
más.
zoom
Looks like a
in?
Bergmania.
Oh,
No,
Marco, can you
Yeah, it's a Bergmania.
wanna in the head,
How to
right?
spell that?
Siphonophore do gênero Bargamia.
Bargmania. Bargmania.
Probably
I-A.
elongata. Look of it.
Is the genus. Wow. You can see the head. It's also known as the
Oh, no,
nectosome.
Maybe not.
maybe not Bargamia.
Yeah, we gotta zoom in a little bit more to find out.
Okay.
Zooming in a little more. What you'll notice is
vemos ahí
that
los
the
neumatóforos,
head,
como
which
se están
is also known
moviendo,
as the nectosome,
son los que
each of the units
permiten
that you see on there are called nectophores.
Don't seem like
They're responsible for
simple
propulsion.
Bag, really.
Bargamia
moving
has like
this
the
animal.
tiny
You'll notice how,
tentacle
like,
nectophore.
Ooh, flexible the whole nectosome is. So we'll
Maybe not
just
Bergmania.
let
Not
it.
Bergmania.
So now
I take it back.
Dougal is taking back Bargmania.
Não é Bargamia.
We'll just
Aqui
let it,
Not a Bergmania.
let it
Coming out.
just come out. Yeah.
The nectophores were more like
estábamos
T-shaped.
viendo los
T-shaped, right.
nectóforos
Yeah.
moviéndose, que son los que les
That was
permiten
really
la propulsión a estos
Could it be?
organismos coloniales
That's
No,
in
it's
a
a lo largo
DPIV.
de la
I thought I
colita
saw
que
some
vemos
brown
ahí
thing.
o el
Did,
resto
um,
del cuerpo
Do
del
we still need
organismo
more
son
4K footage to do the
los
proper
otros
ID?
zooides
Yes.
Dougal?
please.
Okay,
que
so
forman parte de la colonia
It
y
keeps
tienen
coming towards
dentro
us.
de, de esos organismos están los encargados de la alimentación que son los gastrozoides. Entonces, cuando se ven al
what you're seeing
microscopio o a la
is, you know,
lupa,
this animal is
se
really,
pueden
really
ver varios
flexible.
como mini tentáculos
it's-
que son los que se encargan
it's
de
interacting
capturar el alimento, manipularlo y
the water that goes
transportarlo
around
hasta
the
los
vehicle
gastrozoides,
and
que es
or
donde
the
ocurre la
thrusters
digestión.
on the vehicle that's creating some of this disturbance. So we'll sit back and let the water calm a little bit and watch what this animal does.
Belong in that genus, it should be in a different genus that needs to be made and put in a new family.
Okay.
And this is that as well. Look at the way the siphonosome is curling up, like
Uh-huh.
the guy we caught on the first day.
Uh-huh. I'm busy watching the
estamos
nectosome just
ante-
curl up
And that
on
would
itself.
explain why it's not a simple nectar
Excellent.
sac.
Un organismo
And
no
it was, it's
identificado de
slightly
una
wider and then
familia.
goes
They
down.
by
right?
function exit.
Did I do that?
With the,
Ahora no me
They
acuerdo
look
el
like
nombre.
this, yeah.
Mhm.
Hasta ahora ya hemos
But it's different to the one
varios
we caught on the
organismos
first day.
que no están descritos. Así que el profesor Douglas está de fiesta
Animal comprido
prácticamente
que
todos los días.
é construído a partir de vários
I have a
indivíduos que vão se
feeling we are
clonando e
collecting
produzindo a
it, but
colônia.
I
Então
think we
a colônia
we wanna
começa a
do a
crescer da
couple
parte
things
proximal
with it.
ali com o pneumatóforo, aquela parte branca mais brilhante.
Dougal's over here massaging
E
Joel.
vocês podem ver dezenas de nectóforos, que são
What's first, Kakani?
Not Bargmania.
animais
So I think we
parecidos
want to
com
start
águas-vivas,
with
com medusas que
DPIV.
pulsam, né, sincronizadamente,
That's probably all we really
gerando a
want to do.
propulsão do
'Cause
animal.
I really don't want us to lose this animal.
Be attacking me.
Let it
Vamos tentar fazer a coleta desse organismo e também o escaneamento dele através do DPIV.
Ready, Marco?
Yeah, not Bargmania.
Oh, it's coming.
Probably about a meter long.
Ah, lost it. Keep the modes.
There he is.
About a meter, I think.
Oh, the animal?
Um animal aqui, segundo
Lá-
o pessoal, pode ter cerca de 1 metro de comprimento.
Yeah, this is going to be a tricky scan because, I mean, really, we're probably going to be focused on the
Bueno,
nectosome of
Dora,
the
ahí
head.
Renato responde tu pregunta, más o
And
menos
so,
cerca de
in
un
order
metro.
for it to fit in the field of view of the camera, it's going to have to be horizontally oriented.
Vamos a proceder
Ooh,
con la toma de imágenes,
yeah,
con el
right.
scan y después con la colecta.
It's dancing around a lot, unfortunately.
Yeah, the scan is also going to be difficult because the animal is definitely swimming around. For
Oh,
these
it's on pitch.
scans to work really well,
So let's see. Use lower cat. See how it goes.
One thing we could try and do
Acá la
is
colecta o las tomas de imágenes va a ser un poco difícil porque
It's off. Yeah, I think it's not good at all.
el sifónoфoro no para de
Yeah, I can see
moverse
that.
y las corrientes tampoco
Wow.
ayudan mucho.
E é necessário também que ele esteja na horizontal em relação ao feixe de laser, para que pegue a maior parte do corpo do organismo de uma vez só.
lights on. Really close to DPIV.
Should we be better off with the portrait or ramp?
It's relentlessly swimming towards us.
Chunky nectopores, huh?
Yeah. It hasn't shed any zoids, has it?
Not yet. It was a bit
It's
too
almost
close.
unheard
You know, one of the concerns working in Midwater is when an animal
Okay. Lots of.
Hoy nos contaba
gets the
Renato
tail
que
to,
estos organismos
unfortunately.
sifónoforos
That might have been
no
okay.
A little bit
necesariamente
lower.
esta especie, pero
Okay.
en general pueden alcanzar
I think
los
that's in a
4
good
metros de
spot.
largo.
40.
40.
yeah,
40, está no
well,
Guinness Book.
oh, it's a bit to the
40
right,
más
sadly.
todavía.
Sim.
We got a siphosome scan, a tail scan.
Não é essa espécie, não se trata dessa espécie, se trata de uma espécie que chama praia, do gênero praia.
Oh.
Pode chegar, já tem registro de 40 metros, está no Guinness Book. Um outro fato interessante desses animais é que esses nectóforos que vocês podem ver pulsando, ele
Oh.
pode direcionar o jato do pulso para
Nice.
diversas direções,
Think we got a
inclusive
little bit
fazendo
there.
com que o animal nade para trás, né? Então ele direciona a ponta do nectóforo
I mean,
e
maybe.
ele pode fazer curva,
coming
pode
again.
nadar fazendo uma espiral
but it's
e pode
rotating
inclusive nadar para
a
trás.
lot. Doing a lot of things.
Agora, um grande mistério é como que esse organismo se orienta,
It
né?
might
Sendo
happen
que o animal
again.
não tem olho, não tem cérebro, mas
I think
de alguma
he's
forma
stuck
pode
to our
perceber a
housing
sua
right now,
posição
so
Chandelier.
no oceano.
you could see some tentacles that's stuck to the camera on top of. It's fine. It's fine. It might be a little tethered for a
O
while.
Renato não tem receptores ou neuroreceptores nos tentáculos e essas coisas.
It's a shame that.
sistema
Não
nervoso
conhecemos, não sabemos como é a
Yeah, so you
recepção
can see
de,
it. A tentacle is attached
sensação
to the camera.
de pressão ou de sensação
Oh,
química,
Did it
né?
just
Não
separate?
se conhece, né? Um animal que não tem cérebro, então é difícil entender como que esses animais percebem o seu ambiente, né?
Mas ele tem um sistema nervoso assim.
Não tem
starting to
sistema
look
nervoso.
A little
Nada.
bit to the right.
Tem algumas
A little more
células
to the right.
nervosas,
Sim.
algumas concentrações de células nervosas, mas não tem uma centralização, né, dessas células nervosas no que seria
Claro,
um
no
cérebro ou um
Just to the right a little
protocérebro,
bit.
né?
Mas cada organismo seria independente,
Quite in the
assim.
frame, it's just that little bit big, huh?
It's very big.
Sim, cada
This
organismo,
animal's not 40 meters.
na
but-
verdade, eles atuam em conjunto, né? É curioso como que um animal que não tem cérebro consegue coordenar o
Almost.
movimento de vários organismos, vários animais da colônia.
Great. Yes,
Probably
that actually looks
Opa.
really good. All right, so
Bom
we probably want to
escaneamento.
collect it, right, Duke?
No wires?
Uh, just kidding. One more imaging system, and then we'll.
We tried.
Yeah,
Vamos a intentar un escáner más.
yeah. I think we got
I
to.
think it's a bit tired now.
Hopefully.
It's coffee break.
Yeah, look at that. All right.
There we go. All the
So
all the
after
juice.
Joel had to do all of those scans, now he's gonna try and get this siphonophore to be in
60
the
frames per
field of
second.
view.
More data is more better.
Yes, of this other imaging
there we
system
go.
called Iris.
Ooh,
It's looking
good.
really good. Now we just need a head in there.
The nectosome in there.
I think its head is a little bit too close to us. That's why it's that darker reddish color. That's pretty good. And then back
stop
off
swimming?
just a little bit.
Looks like it stopped swimming.
Yeah, which is perfect for this.
It's
Back
also
off a
perfect
little
for
bit.
DPV again.
Nice. Wow. Great. All right,
It seems a
so
little dark. Is it me or does it seem dark on the
I think the
on the nectosome?
I thought it looked pretty good, Yost. It was almost filled completely, and because it's transparent, I don't have high hopes for it.
Change the exposure slightly.
Keep going forward. A little bit more forward.
Aqui então vocês podem ver um escaneamento através do Iris.
Nice,
que é outro tipo de
Green is
técnica
like the sweet
de
spot.
imageamento, né?
Thank you.
Aí,
All right, now we're going to
assim,
do a collection.
conseguimos e agora
Dougal
vamos para a
wants it
coleta.
in the suction sampler?
He didn't.
I don't know. Does
Pudimos
Dougal have a
capturar
preference?
Varias
I know
imágenes.
there's a preference for larvations in the D-samplers, so I say suck it up. If nobody comes in,
Yeah.
Vamos a proceder
let's
con
go.
la colecta.
That's,
No
that's
sabemos.
good, yeah. Suction on.
In the cold.
all right, so we're going to be collecting this animal.
It's tired.
Yeah.
He gave up.
Hopefully.
Certeira. O professor Dougal Lindsay até comentou que pode ser de uma espécie, de
Obrigada,
uma família não descrita ainda.
There we go.
Opa. Sim, conseguimos a
All
coleta.
right, we'll wait for it to settle a little bit and then index, please.
Index.
Yes, please.
Ok.
All right, number 2. Do you remember the name that Dougal gave this? All right.
He said that's undescribed family, maybe Pagese, but he don't give any
Nothing.
any
Address?
identification.
I'm not. Do you know how to spell that? Little fish. Little fish.
Continuando então nossa jornada em aqui mais ou menos na profundidade de 700 metros. Essa é a máxima profundidade planejada para o mergulho de hoje.
There's something there.
Where? How long do you think it's going to take for a Dougal to run in
tem-
here? That pigmentation is so
Muito
cool
interessante
on the
animal.
nectosome.
Tem essa coloração avermelhada com esses pequenos tentáculos
Yeah.
e também com bastante pigmentação no sifossomo, que é a
Uau.
parte final da colônia.
It's like brown pigment on the flat surface of the nectophores. I'm going to look and see if we have anything like this in the deep sea guide. Wow. I think that thing you see to the left is it might be a detached bract from
Esses
either the same
nectóforos
animal
com
or
esse
another one that was really
filamento.
close to us.
O nectóforo é aquela parte que
Let's
está
see.
pulsando, né? É típico do
Phylogenetic
gênero
image
Apolemia.
gallery. Yeah, taking a really close look at the pigment that you see on the nectosome. We have something in the deep sea guide that called Apolemia rubriversa that has some of this kind of color pigmentation, but the pattern is really, really different. Still waiting on Dougal. Wow, if we can fix that focus, that will be amazing.
But sometimes
Eu acho que nós já vimos esse animal
Oh,
em
I-
algum outro
I
mergulho
give up on auto all the time.
e realmente chamou atenção.
Wow, look at that.
Que imagem.
Oh my gosh, that's so
Que imagem.
beautiful. Look at that.
Apenas alguns nectóforos pulsando, o animal está fazendo uma curva, né?
It's got, I can count 8 nectophores. So 8 of those jetting units that you see on its head. You can also see little tentacles, those little red things that are kind of splayed out in between each of those nectophores. It looks like an apolemia to me.
Looks like a Apolemia, but this red tentacle
Yeah,
in
the
the,
red-
in the,
the
nectophore.
pigment.
Yeah, the pigment.
The pigment's really different. And then you can see there's a little float, a gas-filled float at the top. Helps the animal to maintain its vertical orientation. No sign of Dougal, huh?
Um animal fascinante. Tô
Are we
até
just
fazendo
gonna-
Came to
imagens
say
aqui.
that this is really beautiful, in case you guys didn't know.
Wow, these are wonderful images
Maravilhoso
too.
animal.
Do we want to do?
Coloração
This
avermelhada
one, or let's
dos
start with-
nectóforos é algo que é muito raro.
Do you want to
entre
start?
esses
I know I've never seen that.
sifонóforos.
Do we want to start with iris,
Veja
Yost?
que ele está pulsando apenas um
let's
nectóforo,
do
né?
iris.
Então é um tipo de nado em que o animal está fazendo uma curva, nadando na lateral. Agora de longe.
he's gonna do the iris and the idea on this.
Yes, please.
It's not moving too much.
Right? Famous last words.
Beautiful Apolemia.
So beautiful. Siphonophores are quickly becoming my favorite animal. Apolemia. Or apolemia. Little bit high, little bit high. Now down a little bit, and it might be a little too close.
Close.
Okay,
Dancing around.
nice. That's a sweet spot. A little bit further forward.
Did we lose any of the siphonophore?
I don't think so. It looks like, oh, the siphonosome? I don't know. Before we got on it, it looked like it had started to do its exploding act. A little bit close.
Oscillation happening.
A little bit close.
Too close.
It's too close, yeah. Yeah. Starting to get in a good spot. Now it's a little bit far.
Too far.
Yeah, getting these animals, back in the lab intact requires being really, really sensitive. Normally, we have some success with what's called detritus. Those are the two samplers that you see kind of hanging on the front of the beam. Those are essentially a cylinder with two lids that pilots seal them. It seals both the top and bottom lid. With those, we can collect. And what you see on the right is you don't see it now. Maybe a nozzle, a clear nozzle that's sticking out, and that's the sampler. The power that we're using on the pump to suck in the animal. But I will say, some of the animal collections that our team does with the mini ROV at MBARI and other places, they're able to bring back and collect bloody belly comb jellies, which are super, super fragile, using the suction sampler alone. So it just really depends on the conditions, obviously the animal behavior, and how we do the collection.
Yeah, there's probably good. Yeah, straight ahead. That's it.
Wow.
Yeah, do that with
And
RV
the
lights.
animal is not pulsing right now. So what that means is we can get a really good scan without any movement on the animal, which will result in
It's
a
not
higher
swimming now.
quality,
This is a good time.
3D model. And if you're seeing this for the first time, this is an imaging system called Deep PIV.
Oh, there
PIV
it
stands
goes.
for
Nice
particle
swim.
image velocimetry.
Inches
But
of
what you
tail.
care about is that red thing, that there is a laser sheet, and it's being used to illuminate a slice
Firing on all engines
in the
yet,
water. And when an animal contacts that slice, we're seeing basically that cross section. And then along with the laser illumination, we also have a camera, and that's capturing that illumination over time. So all of that data is then put
Orientation's getting
together,
better.
much like a CT or an MRI scan, to create three-dimensional reconstructions
Slow.
of the animal's body.
That's good.
And this works
Couldn't
with
tell from
gelatinous
the movie.
structures, also works with mucus structures. So we're going to be looking for larvacean snot palaces, and we'll be able to do the same thing.
O que a professora Cacani estava dizendo é que esse momento em que o animal está parado é ideal para fazer o escaneamento, para a construção do modelo tridimensional do corpo do animal, como se fosse um scanner, né, um CT,
Whoa.
uma ressonância, um tipo de ressonância que consegue capturar várias
Nice.
profundidades de campo de uma imagem, né? Não é apenas um plano que fica focado, é o corpo do animal inteiro que acaba sendo focado e reconstruído.
Ooh.
Okay.
All right,
we
so
got it.
what are we doing with the animal?
That's a good question.
Next one, please.
Can someone find Google?
Are we soaking it up or?
We might
I
be,
think
but
we
I'm
already
waiting
collect
for-
some of this, but it should be interesting.
You want us to collect it?
Yeah,
Word is gonna stick to everything.
Well,
Word is gonna stick to everything.
well,
Too late.
Too late.
too late,
Suction
baby,
center.
now it's too late.
3,
Right.
safe.
Nice job. All right, so we're waiting for it to come through. Now, I don't know if it's on right now, but there it is. So if we can stop the suction,
Mais um animal coletado.
and we can index now. Great, thank you very much. That was in section
Yay.
sampler 3.
Thank you for the collection.
So far, on all three samples, we've done iris and TPIP. Great. Okay.
Yeah, we're-
Agora,
You're
a
back.
troca da
I'm back.
tripulação.
How's it going?
São
Good.
3 horas cada
So
turno
we're
da
really
tripulação
looking
que faz a
for-
pilotagem do ROV.
Big things. Russ really wants larvations to collect in a D-sampler. And don't mind those tentacles, they're just stuck to the laser housing.
Proof. Life.
Mm-hmm.
Going down or up?
No, we've been kind of spending time in this layer between 600 and 700 meters. So we've actually seen quite a bit. Huh?
Are there rules around this?
Is that a rule I'm not aware of?
Just tradition, I suppose.
I mean, different people do different things. Once we find a depth layer that we think could be a location for a lot of
Uma
animals
pequena
we're looking
medusa
for,
pulsando
so
ali.
we'll go up and down within that layer.
Ketognato. Ah, não, é um peixe. Esse é um peixe, ciclotônia. O ketognato não,
So yeah,
Não-
we're at 700
não nada
meters
dessa
now,
forma. Peixe.
and we're gonna try and search between 600 and 700 meters. So we can like go slowly up.
Frame up or adjust the face or anything? How do you feel?
We've adjusted things a little bit. I mean, this is obviously still grainy. That looks better.
Oh,
And this one looks like somebody hit their head on it.
You forget it's flying. That's what you're worried about.
Yeah, I know. We write down every day. So I,
Where are the calyxes?
They turn them on the lights page, but to actually bring them up, you have to adjust them there.
Change the power.
Yeah.
Where are they mounted on the vehicle?
There's two kind of midway on the headache rack. Headache rack and out, and then there's a central one kind of pointing straight down.
Okay. All right. We are searching again for
Outro
denizens
usuário
of the
aqui,
deep.
risária. Passando pela tela. Outra risária passando aqui embaixo.
There's little copepod. It's a lot of copepods. You see particles. Ooh, what's straight ahead? Oh, that is a
peixe
little fishy.
bonito.
And then to the right
Oh,
is a
Tem uma
larvation.
casa de larváceo ali.
Pretty small, so we'll see if that's something we can look at and maybe collect for Russ. I know he's itching for more.
Então há vários especialistas em vários grupos de organismos do zooplâncton,
is.
principalmente.
If we can zoom in and see if there's
E
an animal in
cada especialista tem um interesse por um tipo de grupo de animal, né? Então esse aí que é uma casa de um larváceo,
there. When she
me
comes
parece que está vazia. O animal não está na sua casa, né? É um animal que produz continuamente essas casas. À medida que essas casas
slow and steady.
se entopem de partículas, ele deixa a casa
It's really
e
small.
sai nadando e passa a produzir uma outra. uma outra casa. E essas casas têm um papel fundamental no
In case you're wondering, this is a
processo de afundamento de
catalyze.
partículas, né? Uma vez que elas acabam sendo habitadas por outros organismos que colonizam essas casas, né? Principalmente crustáceos, copépodos, e acabam afundando com rapidez, né? E levando matéria orgânica da superfície para o fundo dos oceanos, abastecendo as assembleias, né? Os ecossistemas do fundo dos oceanos. Então, vocês podem ver, tem pontinhos brancos ali na casa. Provavelmente são pequenos crustáceos que vão se agregando e se alimentando, né, das partículas que aderem à casa.
Yeah, we're just trying to figure out if you can see the animal in that mucus house. And if you haven't seen this animal before or know what it is, don't worry, not a lot of people do. These are larvaceans, also known as appendicularians. They are tunicates, basal chordates. They do this awesome thing though where they secrete mucus from cells lining their head and are somehow able to blow up these mucus structures that you see, much like a balloon. And I see the animal. I think the head is that
Opa, o
longish
animal tá aqui
white
assim.
thing,
animal
and the
tá dentro
tail
da
I
sua
saw really
casa.
briefly like a hint of blue, like a blue shimmer. So if we can collect that
Like that.
in a D-sampler,
All right, zoom out.
Are we collecting that one?
that
The
would
one with the fingers.
that would be a D-sampler. I don't think this is the one with the blobs. I think this is.
Vamos tentar fazer a coleta desse animal aqui, então.
I'm a
Muito
little worried
delicado.
because it's so small. I'm not going to be able to see if the
Pequeno e
animal
delicado.
escapes. So we really don't want to collect this if the animal's gone. I think the animal
Olha
is
toda a
still
estrutura
there.
da casa, aquelas duas esferas, na verdade, com formato mais alongado, são as regiões em que o animal adere partículas, né? E a partir da aderência dessas partículas, ele
Yeah, I
secreta
think it's still in
um
there.
muco e depois puxa o muco, que vai aderindo partículas e engole. É como se o catarro do animal, né, servisse de alimento depois que aderiu muitas partículas, né? Então, é uma estratégia de alimentação bastante interessante que esse organismo possui.
This looks so small. There's also, is that the same thing we're looking at? Or is that a, okay. If we can get it in a D-sampler,
I'm trying to like pork chop too.
that will help. That will help for sure. And we can find Russ and he can take a look at some of these images and then we can dump it if it's not unlike
To see what this is. What would we really want to know if we need to collect it?
Well, yeah, if we're gonna collect it anyway, but it'd be good for him to look at the photos just to see if this is something we want to commit a D-sampler to when we recover. That's the nice thing about D-samplers is, you know, if you come across another animal that is more of a higher priority, you can drop the animal that's currently in it and replace it.
Yeah, we can get number 5, thinking?
Which one do you want to go on?
4 is that one.
You want to go on
It's the
5?
outside one. Alright.
Off to the right. There it is. I don't know where that is.
I'm here.
Russ,
Oh, Russ is here.
is this something we're gonna try and collect it. It looks like a small oikoplura, but it doesn't have the bulbs.
I was trying to see where it's at.
Oh, let's just watch, make sure it doesn't.
Still there
He is there, but I don't know if this is, do you want this?
for the moment.
All right,
He's there.
it's still there.
Speculate. There he goes.
I think he's still in
I
there.
was just waiting for the current to hit.
It's still in there.
There he is.
no,
He's
no.
still close to it.
No, yes,
But he's
now
out.
he's out.
No.
Yeah. And I can plur it.
Do you have, the oikopleura was collected in the D. You got footage of that? Right?
Which from the last dive?
From the last time?
I believe so, yeah.
Okay. Because it was a Mies oikopleura.
I wasn't in this room when that
No,
was
I'm just
collected.
saying, we have, if we
That's
have,
great.
if we have the house, we now have the specimen to confirm exactly.
All right, well, I'll ask you what we got.
Yep, I'm sure we always zoomed in before we
This is a
test them.
shame. Okay.
So I may not have, A roll of it, but we should have at least B roll.
Okay, that's exciting.
So.
Are you
Yeah, and
excited?
It was in pretty much perfect shape, too small to image, but, Manu put it in his system so we can
Onward.
reconstruct 3D.
Nice.
Okay.
All right. Try it again. So. I see something dark in the background, but I don't know if that's something on the camera.
Peixinho,
Yeah, that
Lá
looks
pra
like a
baixo.
jelly.
Yeah, it is.
pequena medusa.
Going.
Well, we can look at it real quick and then.
Vamos dar uma
Move
olhadinha mais de
on.
perto nessa medusa.
I know that catches my eye too.
Yeah. Wow.
Uma narcomedusa com 4 tentáculos. Pode ser do gênero Aegina, Aeginura ou
Any guesses?
vamos fazer aqui o registro desse animal. Ou Baticorus também. Olhar de perto.
An Aegina of some type,
Ou
maybe?
Baticorus, maybe, or Aegina. Yeah.
I haven't seen the tentacles kind of
Those tentacles are beautiful.
in a bent at an angle like that.
Almost looks like a macrophage.
Right?
Yeah. How is it?
Paging Dougal Lindsay.
It's the same Bathychorus as before.
Pode ser Bathychorus, se tiver ali, tem que dar um.
Maybe.
We should see in the margin.
That one yesterday had his tentacles in his mouth. It's weird.
This is so interesting.
Cannot see it.
Let's just get close. Get close.
You snooze, you lose. Well, we got some really nice shots of it. Let's go and
Posição de tentáculo, parece um helicóptero esse animal, né? O tentáculo vai na frente da água-viva e isso é típico dessas águas-vivas do grupo de narcomedusae, né? É um animal que deixa o tentáculo na frente e isso pode ajudá-lo a capturar alimento, né? Uma vez que a primeira parte do corpo chega é o tentáculo.
all right.
Então o animal não gera aquela turbulência que pode afastar animais e se aproxima de uma forma silenciosa das suas presas, né?
I don't know
Então,
how to spell that.
essa é uma estratégia bem interessante que essas espécies de narcomedusa possuem.
H-Y-K-O-R-U-S.
It's not in the deep sea either.
Not everything's.
I know.
No.
The jelly daddy is very busy working on jellies next door. So
Well,
reduced availability for us.
potentially Bathychorus. We did, I think, collect a few of them yesterday.
During emergencies,
So
Oh,
he called himself that. So we're
Oh,
not
okay,
doing
fine.
that.
Oh, that sounds like that. Line up the jelly daddy signal.
All right.
The jelly father.
I like that. It's like very formal, almost biblical.
All right. Thank you very much.
Mais uma.
I think he
Let's
doesn't even really need a
let's keep
signal.
going.
He
then.
comes
Yeah.
running in. Something special.
Yesterday he did fall on the wall and he
Oh, he's
did
got one eye on the screen.
Yeah, I think we had a Bathychorus chorus yesterday. So
Yeah, it seems to be Bathychorus. I agree. Yeah. Narcomedusae, Bathychorus. Okay.
Yeah, I already put on.
yeah, Jen. That pretty much describes all of us.
Bathychorus. No.
All right, what are we gonna find now? Any guesses? Someone wants a cephalopod. Lakshmi wants to see a
They're
cephalopod.
so cute.
Uh,
Cats of the ocean. Maybe mammal looking, but I guess we're too
Vampire squid or
but we're gonna have to be very quiet about it because
Dipstaria medusarum, also a
I
great.
don't know if I want
scyphozoa.
to collect that.
Oh,
I know.
Perhaps
I will leave the room.
Temos uma lista de desejos de animais muito grandes, fantásticos, bizarros das profundezas que nós queremos ver. Estamos discutindo isso. É uma lula vampiro, se é uma água-viva gigante, se é um grande
Yeah,
siphonophore.
something happens,
Tem grandes
and
possibilidades.
then maybe like button, you hit a button. And the
Olha,
lid just goes.
hold on,
That's another jelly. That
Another
might
one.
be a similar. Oh,
Seems
I
to
would be into
be-
a deep star. Yeah, we don't see those a lot.
Oh,
That's
seems
this-
to be a
that looks
torella.
like-
Running out of trash
Looks
bags
like
here on
that
the
a
ship.
torella we
That
are
looks very
collecting.
similar to the jelly we
A torella
just
we
saw.
saw yesterday and the other days. Maybe
what is that?
This is
take a close look.
Agenora.
Or
For your next drink, spell that.
I think Aeginura. Its
No, no, no.
tentacles look
Yeah.
a little
People
bit
say
different.
Agenura.
Yeah.
It says Agina for Agina.
So you need to tell him how inconsistent his pronunciation is.
vamos ver de perto. O animal já
All
não
right,
está pulsando.
so a lot of requests in the chat.
Não é
I
uma
mean, we
siphozoa,
already
ela é
got
uma hidrozoa
scans
mesmo.
and a collection of this animal, so we probably don't need to.
Opa, já chegou aqui.
We've summoned
The Jelly Daddy signal has worked.
Ok.
Dougal.
Can you?
Trying.
You've already zoomed in, haven't you?
No, not really.
I mean,
Still trying to get him up to where he's not.
Whatever it's been eaten a lot. It doesn't have many tentacles.
so Dougal, what's the difference between Aegina and Aeginura?
Aeginura,
Besides
lots of
the way you
things.
pronounce the I. There's a question. There's a
Aeginura's
question.
got 8 tentacles for a start, and it's kind of transparent on the top and brownish, kind of like this, except the whole bottom is brownish usually, not just the stomach lining like we're seeing here.
So this is not.
So this is
not Aeginura.
different.
I think this might be, well, I don't want to call it yet, but it looks like Aegina brunea.
Why do you say Aegina when you say
I put it in a new
Aeginura?
genus in 2017. So
So you want to collect this too?
yeah,
What did you say it's called again?
Aeginona
Aeginona.
brunea, and if we can get it from the side so that we can see where the tentacle roots are.
Aeginona. Olha só.
Aeginona
Brunea.
brunea. And I've only ever caught one in pretty bad
Essa é
condition.
uma aula de zoologia aqui, de identificação
So
de
someone clearly wants to
narcomedusa.
No,
collect
it's not.
this jelly.
It's not. I take it back.
What is it?
The
Não. Volta.
wait.
Muito pequeno
Fix that
animal, deve ter
focus,
um ou 2 centímetros.
Take a look at the tentacles. They
Little
seem to
close.
be aligned with the
Stay away.
stomach pouch
Alright, let me
then,
settle
rather
down.
than between.
Yeah, just let it.
Wow.
Is it between?
I know, Jen.
or
That's
is
how
it-
I feel too.
There's a couple in between
For all we
each.
know, you
Isn't
could
there?
just be making it up on the spot.
É quase o tamanho
But
de
he's-
copépode.
He's the expert. I'm not going to
Muito
question.
pequeno.
This is the Aeginona and the Aeginona.
Nope. Dougal, you have enough info?
No, I'm sorry, not yet.
Oh, okay.
It's an
Well,
Aeginona.
Because it's been damaged, it's been eaten and it's regrowing, and there's a couple of weird things happening in there with what's regrowing.
Right.
Yeah.
Bored all the time.
And the other way to know if it was an Aeginura would be to look at the statuses, but you can't really do that from this distance.
Set some stuff. Let's freak out.
whoa. That was amazing.
Muito difícil dar o zoom, um animal muito pequeno.
Uh-oh.
Yep.
And yes, I do believe Dougal has a brain.
I know.
You do believe
But does it have a brain or not?
There's a question in the chat.
What is it missing?
There it is. Oh, there's another Physonex siphonophore.
Did we lose it?
Ok, deixamos ela. Let's collect the
Dougal,
small one.
Oh, that's a doliolid nurse.
Ah, uma nurse. Não é
The guy with the long tail. You see, when it swims, one pulse takes it a long distance. If it was a siphonophore,
Mhm.
it'd have multiple pulses and it
Yeah,
wouldn't be
yeah,
moving
yeah, yeah.
quite so fast each time, and like jets.
Yeah. Here, just leaving it.
Oh, there's the dolly.
Oh, there is a doliolid across the front porch of the vehicle. So Dougal,
Well,
we might
maybe we might just have to catch it. Yeah. And I'll
We're gonna
sort
have a
it
busy
out
day.
later.
because it might not settle. You might want to catch it.
'Cause
I
without
don't
looking
know
at the
what I
statuses,
said.
It's hard to call.
It's really small. But we think the he refers to you, Dougal.
To the Aeginura or to the Dougal?
It's so pretty.
Kind of.
I know, both maybe. Kind of.
Ele tá
This
com
ambiguity is not good for anyone.
Ó. Tá com um tentáculo provavelmente comido, né, por algum outro animal.
Interradial, right? Except there's also interradial some
It
pouches.
looks like someone snipped its
Oh, maybe they're
tentacles open.
split.
Yeah.
Seen some things.
It looks like some of his tentacles have been eaten.
Lappets.
So Dougal, do we want to collect it?
Hola Pablo,
Could be.
justo tuve que salir porque
Yeah.
todavía estamos
How many canisters
procesando
you got?
unas muestras
The whole lot,
de,
right?
Have you been taking notes?
de las redes.
We are on the board.
Yeah.
Tuve que salir porque estábamos procesando todavía unas muestras de las redes de hoy a la mañana y bueno, me llamaron y salí corriendo. Justo ahora estamos yendo atrás de esta pequeña medusa que creo que es parecida a la que habíamos visto antes y lo que estábamos riéndonos era que no entendíamos la pregunta de Christian si a quién se refería, si tenía o no cerebro. Así que nos estábamos riendo acá, si era de Kakani o de Dougal. Y mi respuesta fue de que tal vez podría tener o tal vez no. A esta altura, a esta hora, hace tantos días, no, ya no sé si no fueron tantos, pero como estamos casi sin dormir, parecen muchos más.
He apparently has a brain.
Yeah, I think that they are talking about you or the shellfish.
This? Oh. I am a wannabe biologist.
Las
I
medusas
don't pretend to
no
claim
tienen
that I am.
un sistema
I'm
nervioso
learning all the
central,
time.
tienen células nerviosas distribuidas y algunas concentradas en otras partes que les permiten el movimiento.
All right. I mean, the other thing is I have a little more familiarity with what we see in Monterey Bay, but that represents such a small place in our giant ocean. We're learning a lot as we go. Yeah.
We're like a bit up, so it's sort of the same level as your...
Maybe I can swap with you and then you can have a look at it.
Do you really want to hear me?
Tal vez en
I think I
algún otro momento
broke it.
de la noche de la transmisión tenga que salir corriendo de vuelta para seguir procesando muestras. El ROV va a estar en el agua hasta las 2 de la mañana, me parece, si no estoy equivocada. Después, así que llega, ya largamos las redes y a las 6 de la mañana se recolectan, se recogen y empieza todo de vuelta. La primera transmisión de mañana va a ser a partir de las 11 de la mañana. Así que estén atentos los que son diurnos y nos acompañan para más traducciones o para seguir viendo las maravillas que estamos encontrando en la columna de agua.
To the left, I see a little bit of mucus. Wait, it's all right. We probably passed it. Never mind.
Left,
To the
center.
left,
Oh, yeah.
do you see that? Nice. What is this? I know, I was going to say, that's very nice of you to say that. I will take it as a compliment. Straight ahead. What is this? That looks like a siphonophore.
No,
It's a siphonophore. I got that before Dougal.
it looks like a Foscalia maybe. It's dropping pieces of itself pretty quickly.
It looks like it's dropping zooids.
Yeah,
Is that a
Forscalia.
Forscalia? We haven't gotten a lot of good scans on
And there's also
Forscalia.
Mandela bispectaculata as well. Can you capture?
Oh, we didn't decide this one.
Ooh, look at that.
Just capture once while we.
Look at that beautiful color.
It's definitely a Forscalia.
Yeah. Do you know the species?
It looks a bit like Asymmetrica, although the nectar zone is much fatter. This would be a good one for you.
We would love to get a scan.
We have some from the past. It was always very hard to get good scans because they always.
Because they move.
Lindo, siphonophore,
and if not, we
gênero
could do
Forscalia.
iris.
Esse ele tem os
Do you want to start with iris, yes?
nectóforos colocados
Do you want to start with iris?
situados de forma assimétrica. Eles não estão no mesmo plano.
Yes.
All right. Iris, please. Iris.
Sarah, you warmed up?
Ó, um animal que ele
Let's do it.
provavelmente em reação à luz do ROV começa a se desintegrar. Ele solta, deixa para trás um
Forscalia,
rastro
which
dos tentáculos
defies
do
another
sifossomo,
siphonophore.
que é a
Yeah.
região
Oh, yeah.
que porta os tentáculos, e os gastrozoides, né? Os zoides responsáveis pela alimentação e os gonozoides também responsáveis pela reprodução do animal. Então muitos desses organismos deixam esse rastro de tentáculos, né? Acabam se desintegrando como uma resposta à luz do ROV.
Bring up the iris cameras.
Jimbo, can you pull up the iris feed? Oh,
Forscalia.
never mind. We got it.
Iris is being done for that, right?
Yeah.
I kept the depth limits the same as before, so 50 to 300 should be good.
All right. Here we go. It's going in iris. Do you see it in your...
What's the reason for you to decide if it's iris or...
Find in it first.
We like to try both, but given animal looks like it.
There it is. It's a little, it's right in your viewfinder. Now it's a little bit higher.
Really transparent to animals, and iris is better.
A little bit higher.
Capacity to it. In this
And
case,
there it is. So now you want to go further forward. Oop, and to the right.
and then if
Because he's out of the
iris
camera
is
now.
essentially 4D information, right?
Yeah, yeah.
3D
Yeah,
on
sometimes
every
they
frame
have
at 60 frames per second. Whereas with
Yeah.
DPID, we rely on scanning the animal to complete the 3D
Or
spatial.
report.
So ideally for that, the animal isn't moving and there are no currents and nothing else changes while we do the scan. It's like if you ever got a CT scan or an MRI
Oh,
scan,
right.
they tell you to sit still. I wish I could tell these animals to just sit still.
Yeah.
Now, okay, it's a little high.
Don't have this power yet.
Now to the right.
But I think these pilots are working.
And
Yeah.
that's the
It just
right
like sits still
distance.
relative to the vehicle.
Oop,
So it
to the
moves
left a little more.
moves along with it.
Yeah.
A little higher. That's really good distance.
Alright, we have
They
one more
they
80
are
stars.
super maneuverable, so our best hope is for them
It is
not to
captured on
swim.
DPIV, but like not during these.
So now they're going
scans.
down.
So iris we do because you see that whole particle field, right?
Yeah.
We got a really amazing scan with DPIV on that glass squid last night.
Probably do is particle tracking.
And Yost did a preliminary 3D model, and it does look really good.
I mean, there's some automated methods, but like it's
Look close.
individual particles.
Yeah, yeah, I see, I see.
PTV rather than
But would be like a two-dimensional
Oop.
particle tracking or three-dimensional?
Right.
Because each of the particles here that have color,
Yeah,
they
yeah.
have 3D information.
Yes, this is iris.
You actually look at the 3D model,
Mhm.
you see these particles flying around in space.
Oop, there it is. It's a little
Mango
close.
is more on morphology.
To the right and a little close.
For that, I think organisms are a bit more suitable. They don't run away as fast. And we can set the vehicle down, take time to line things up perfectly. We've done that with sponges. We also work with octopus.
Yeah, you saw the paper publicly from octopus.
And thanks to the support of the Susakawa Foundation, we've been able to spend time developing improvements to the software that we use to collect this data and generate models. And so I've been really happy with the improvements that we've made. It's been making it much easier for us to collect data on the fly in a semi-automated fashion. It's a little close. Oops. A little close. Oh.
It helps if you lean over.
Yeah.
Your shoulder needs to move too.
Yeah, lean into it. Ooh. A little forward.
I do think we got some there.
I think we did for sure. Let's get one more good one. Oh.
Got multiple decent passes. Should we move to the UAV?
Yeah. You ready for the next fun challenge?
Always.
We want to do.
Pilots do have a preference of which one goes before the other.
Yeah, I'd be curious.
None of them.
Wow. Wow.
Beautiful.
A little further away.
Further away?
You want to move further away?
Can we go to 100? Can we go past that?
Yeah, the warm colors is close.
Ooh, that's too far.
Did I just see a Tuscadarian
What
on
is
the
happening?
side?
Oh, yeah.
Yo,
Did not go to 100%.
Did we?
What happened?
Yes?
Now we know.
Oh,
Not
okay, that's a little
Oh, no,
close,
not
I
a
think.
Tuscadarian.
Not sure.
Only one of the
Hard to
coolest
say.
I can let you
siphonophores.
go whenever you want.
Yeah. I will just.
Would you like to?
I would just do the
Yeah.
A
last.
little bit
Give me
lower, I
10
think.
seconds. I'll be right back.
There it is. To the right. Oh, a little bit too close. Yeah, the volume that we're trying to target here is, what is it? 20 centimeters by 20 centimeters by 10
Force
centimeters. This is not easy.
General force scale.
All right, thank you so much.
Buenas noches otra vez.
Let's do it.
Lo que
Let's do
estamos
GPIV
escaneando
while we
ahora
can.
es otro sifónophoro.
Next. And you have to tell us what you perhaps
del género
prefer doing.
Falkor. Si voy y vengo, es porque todavía estoy haciendo cosas en el laboratorio.
This one was not that small.
Así que cuando siempre necesiten alguna traducción específica o que les cuenten algo, pueden pedir ahí en el chat, que siempre alguien de acá del control da un grito y nos llama tanto a mí como a Silvina.
He came back and he said you'd lost it.
Or the Icopleura?
What did we lose? What did we lose? Is that what
Oh,
you-
he said he got Tuscalora. He said for Scalia. Is that what he was saying?
Yeah.
Okay. I can take over at any point if you want.
Focus.
Now that I've got everything kind of held.
Terrible. That's just too.
You've been on for 2 hours, right?
Sure.
You've been on so long?
Hi, Kakani.
Quien entra al comando ahora de la inversión es Ross. Sí, Renato ahora está políglota. Hoy también estuvo hablando un poco de
Nothing
japonés.
collected?
Como tenemos gente.
Since the siphonophore. About half an hour ago.
Solar, uma bateria.
Estamos falando que teu espanhol é muito bom.
Rotation happening. These are so tough because they're not quite symmetric and they always tend to rotate a little bit. See if we can get lucky.
That's a little out.
That's crazy how they can just hang.
Yeah.
How do they know that they're hanging still?
I'm not sure.
Oh,
Well,
you
that's
wanna
a deep
you
goodbye.
wanna be scanned. All right.
I've done some scans of cooking.
Yeah.
Which
Well, now you have a free one.
You really?
Do it. Do it.
Despite they're not transparent.
Yeah, if they're small.
Ah, I see.
No, too far.
That's cool.
Bom escaneamento.
Come up, dude.
I couldn't tell, it was still like rotating.
Uhum.
You'll be staring at a lot of this imagery later. Oh, look, synchronous pulsing. It's trying to go somewhere.
One more.
Ooh,
Yeah, it was rotating too much there.
really?
Yeah, because you see the
Yeah,
whole,
yeah, yeah.
sort of their axis is doing
Uh-huh.
this,
Yeah.
and then you're, like it gets exaggerated.
Yeah, but I understand that should be horizontal in relation to the plane, or vertical.
Yeah, so if you have the laser shoot like this, and the animal's moving like this through it,
Mm.
you can get some really strange artifacts.
Oh,
I do not yet have a way to
I see.
Qué lindo.
Qué lindo. Boa.
It's technically possible, but we haven't had much success. I'm gonna zoom a little bit because
Estou conversando aqui com cientistas, né, com o Yost.
Para el final del crucero, todos van a terminar hablando español o portugués.
O cientista
Esperamos
Yost Daniels,
que
que é
así
do
sea, al
Embare
menos
também,
algunas palabras.
e ele comenta que há uma postura correta, né, mais ideal para que o animal passe pelo plano do laser, né, e que tenha um escaneamento mais limpo, né? Então, se o animal estiver rodando, se ele estiver numa rotação, o escaneamento não fica muito bom. Então, essa última passada foi muito boa. Agora, mais uma
Are you
tentativa
sleeping at some point?
de que o animal passe para que se tenha um
Yeah, I do.
melhor modelo tridimensional do corpo do
I-
animal.
Always
I
see
think
you
I get more
awake.
sleep than a lot of people.
Oh.
We might have a very similar schedule.
Yeah, you are always awake. Every time that I see you, Kakani is
Dougal
here.
is always awake.
Yeah, Dougal is always
I
awake.
think he gets at least 3 more hours, or fewer hours of sleep than I do.
synchronous pulsing.
Ooh, that was pretty good.
A lot of rotation.
I think everything we're seeing is a camera interaction.
Yes. Yes.
Alright. Feeling about this. And no
Are
pulsing.
we collecting this after the scanning?
Oh,
Yes.
Oh, I think that's why we're doing it. Yeah, siphonophores are quickly becoming one of my favorites. Do you like siphonophores?
Yes, now yes, but I'm preferring to collect copepods or raiserian. They are
Raiserian.
well,
Oh,
You-
wow.
they are,
So
they
much
are
work.
not very
We have
representative.
contractors helping.
Oh, in the literature, in our like
3D models.
understanding.
Yeah.
Yeah.
Usually you could spend 30,
Yeah,
35
they want
hours
to know
per
about
model.
what you're studying.
Adam's on the chat. Jamie's
Bueno,
up there.
Pablo,
He's, his data's gonna be any good
Esta
for
campaña, lo que el objetivo principal son los estudios de la biodiversidad marina en la columna de agua, principalmente de los organismos mesopelágicos, que van desde los 200 hasta los 1200 metros de profundidad. Estas regiones están poco descriptas y poco estudiadas, entonces a lo largo de este crucero fuimos descubriendo entre comillas algunas especies que nunca han sido descritas. Además de eso, los investigadores tanto Cacani como Just del Instituto Monterrey, siempre me olvido el nombre, equiparon el ROV con una serie de cámaras que permiten el escaneo de los organismos para formar imágenes tridimensionales o 4D, y además permite estudiar los movimientos a través de las corrientes que generan estos organismos cuando se están moviendo. Esto son todas prospecciones así con finalidades de ingeniería. Entonces lo que ellos hacen es, una vez que tienen estos movimientos, colectan estas informaciones, estas imágenes, usan esa información para futuras creaciones robóticas.
Sarah, what do you think? Did we get the best we're going to get from this animal?
Out of frame. Was that out of frame?
Sí, a estas profundidades no llega la
Need
luz
to have
solar.
like,
La luz solar no
What-
llega acá en estas aguas.
what we need is an overlay
oligotrópicas, es
that
decir,
kind of at zoomed out, that has a mark there and a mark of where the far side is, like a piece of plastic we can just drop down.
a estas profundidades no llega la luz solar. En estas regiones oligotróficas los rayos solares pueden alcanzar tal vez 50, 100 metros, capaz un poco menos. Entonces, como vemos y como estuvimos viendo hasta ahora, una vez que... me perdí mirando el siphonophore. A estas profundidades, la mayoría de los alimentos, la mayoría de los organismos se alimenta de los que son filtradores de la nieve marina, o algunos otros son predadores. Entonces es muy difícil para nosotros acceder a estudiar estas especies.
There's so much we missed when we're trying to look at
So
three
we got three samplers, and this one we were just doing.
Okay.
Assessing rotation.
All right, so we got 5 samplers emptying both these. Let me just mark it off on my copy.
Oh, yeah.
All
So
right.
which, I
Yes.
can't read this low light.
Alright.
So
Let
yeah.
me just mark it,
How you collect them?
because I don't have my glasses and
La
it's low
idea
light.
básicamente, así a grandes rasgos,
Russ,
es
are we
que
collecting this?
estudiando los movimientos y la
I don't think we
distribución de
collected a
estos
4-scale
organismos
yet, have we?
con estas imágenes tridimensionales,
We've seen them shallow, but we haven't seen them at collectible.
los investigadores
Let me
del
check with
Instituto
Dougal while you're still set up. if you want.
de Monterrey pueden o les permite diseñar, estoy pensando de la mejor forma de explicarlo,
Yeah, so it's actually
desde
pretty
donde
quick
yo
to get a
entendí,
quick overview. I've done a
les
whole
permite
bunch here. I can
diseñar
show you some volume renders.
o los ayuda a diseñar
Yes,
los
correct.
tipos de robots con
May we?
diferentes propulsiones inspiradas en los movimientos de los organismos
Suck it up,
marinos.
please.
No sé si ahí quedó un poco más clara mi explicación.
I hope just the ticks are enough.
Puedo informarlo
Yeah, just-
mejor y
just the tick.
explicar
Yeah, it's on this
mejor
side, so
después,
yeah.
más
Oh, good,
tarde.
okay.
Yeah, I just take the first three. Yeah.
All I just need to be able to see it easily.
Sorry, yeah.
And are we
especially
doing
this,
any, no distances, Chris? Or this
No,
type?
there's no Niskins at least. this
All right,
time.
I'm just
Yeah.
making it darker. It was done in a really light picture.
Yeah.
So each scan, like a good scan, is only like 3 seconds long or something like that, because that gives us almost 200 frames, which is sufficient. It doesn't mean, usually the spacing between the frames is a little bit larger on the pixel resolution, so we have larger spacing in Z than in X and Y, usually by a factor of 2 or 3.
There's another siphonophore out there
Cualquier
just
cosa,
teasing
Pablo,
us.
puedes buscar más información
It's giving us
en el
the-
Instagram o en la
Well,
página
we're after the other one.
Mbari, con B
But with
larga.
multiple asymmetric reoriented, so it's at the same time.
MBARI. Es
Different one for all.
la abreviación del instituto
Not just for us.
For everyone.
y ahí explican bien todo lo que ellos hacen.
You did the ID.
What's up? What about?
My PhD was in laser-based particle accelerator.
I don't know what it is. You looked like you had that perfectly lined up, and yet it still ended up.
It's kind of like this with lasers.
Not related to
Not underwater, not
Oh, when did you get
He's
into
in
this?
a lab with 3-foot thick walls, lots of concrete, lots of lead. I never got seasick though.
But when did you get into this then?
Immediately after my PhD.
Oh, so
actually.
you're
Well,
postdoc,
so I
okay.
took some classes in marine science during my PhD. My PhD actually came without any classes. Was the Dutch version of PhD is the class with. That's part of my master's degree I did.
Yeah. You just take a look.
Oh, I'm telling everyone this?
Can the scan data be combined? I think the answer is yes, but I'll let you answer.
Yeah.
Scans.
All right, so can the scan data be combined?
Yeah.
On rare
Oh.
occasions, we have
You got a
done
piece
that.
of it. You got some of the
But
tentacles,
it's incredibly difficult actually because there's always slight differences, especially
and you
for
got
these,
you just got
moving
some
animals.
some of the nectophore.
So it's kind of tough. We usually just go for one perfect scan if we can manage. Sometimes different body parts of an animal we might combine. We dream to combine them.
Traduciendo un poco lo que decía Just sobre la pregunta de si las escáneres se pueden combinar o necesitan una imagen. La respuesta es que se necesita una imagen perfecta, básicamente, pero para eso toman varias imágenes porque obviamente los organismos se están moviendo constantemente en la columna de agua, no solo por las corrientes, sino también por el propio movimiento del sifónoforo.
There he is.
Cool.
So if you give it like a couple second pump and then give it a few seconds.
We need to preserve the magenta. So if you want to have a look at under the
Yeah.
scope now. Thanks. Under my scope, yeah, and then John will take it. Okay. I've taken photos.
Then I pass the drone.
Yeah.
Go direct.
Yes.
Where's John?
You are free now.
You are free. You're like
Free lady.
an orca. Willy. Willy, you're free.
Hello! Long time no see. Yes. I keep discovering there's all these people on this ship I've never met before.
Don't you get enough for me today?
Oh, yes. I would like to collect a few more of those holy opposites that little chain of animals. That is so cool.
copepod, a very big one.
And the big RV
Oh yeah.
probably for aquarium.
Look at it swim.
I haven't seen one yet today.
And a very beautiful, very rare jellyfish.
Stop jellyfish. all the trash,
You
all
think
the
Filaria?
bags are collecting your jellyfish. It's an oiko, maybe?
Oik, yeah. Oh, he stopped swimming. Why would he stop?
You're right over a D sampler, you want to put it in, go ahead. Get their D open.
Yeah.
Look like they're perfectly set up. Oh, close it,
Close, 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 close. Ah,
He's out, he's right there.
Oh.
You waited too long.
Did you go through the bottom?
Oh, you got out.
Yeah.
Bummer.
Escape. Again, another one that is free
Hmm.
tonight.
Read Willie again.
Be free.
Be free.
Mm.
Free.
Free the larvations.
Speak to the others. Tell them. Tell them.
Good catch though.
Right there, though, that was.
It was just too late on the trigger.
I have to take the safety off first.
Okay. Your turn to flash.
Leaving the safety
Yeah,
on.
it is.
It's not much fun.
All right. Suni likes a challenge, so let's set one up for her. Nothing over 5 millimeters in diameter.
Now we're talking.
Copepod?
Copepod.
Marine snow, marine snow, marine snow. I can do more marine snows than you can do, Copenhagen. In
Yeah.
fact, because
That's
from
all.
the US, I'm gonna do an acronym. I'm just gonna say SM, SM, SM.
And wave that stick at it.
No, marine snow, it doesn't. MS. Okay.
There was a brain part.
It's it's the Pagantha House Scaredoos. The missing link.
The missing link. I thought that came from Africa.
Naka Medusa?
And is that a small Batrachoris in front of it? Keep going.
There's a copepod.
No, it's a copepod. Dang.
Maybe we should look at the
I thought
copepod.
it was. You've got this now collected, right?
No.
No?
I got it from the Gulf of Alaska.
That's a long way away.
Heavy swimming.
Where are we? About halfway through down?
Is that big copepod again?
That's the same copepod. Yeah. The name is George.
Jin. Oh.
Really? You saw its fifth leg?
It's like Wendy to me.
David, what triggers you to study copepods?
What triggered me?
Yes.
Nobody, the other communities wouldn't accept me into their group. It was the only one left.
So why aren't you a physicist then?
Right. Sitting in a dark room alone.
Ooh, a nice shirt.
Thank you.
That's what the
Thank you
Discorders wish
so
they could
much.
have. Yeah.
Russ, is it our floor or am I?
Try later.
I was trying to come back into my rotation and I missed first house.
Okay. When did we go?
So you still got stuff they're trying to get imaged, or are you all done?
Nope, all done for now until the System X ready.
Philippe started to disintegrate under my microscope, so he's going into genetics.
Got it.
So you're all
So I'm
done?
all done. But if you'd like to stay up there, that's all good.
I'll stay up.
Okay.
Yeehaw.
Got a few more elevations to snatch up from. More out there.
It's the same dude.
Whoa!
Are we finished on this, or are we collecting?
Strobe in the red light.
Can we collect?
No.
We have 4 canisters left, 3 that we can close.
How many jellies we got already? I don't want to be a hog.
How many gels?
How many
Do we
jellies?
have other stuff?
We have siphonophore.
Okay,
Really?
you've got.
No,
yeah, you
I got
don't. You
enough
don't
to
need
do.
it. You're too busy.
I got enough to do. Yeah. All right. You got
Only
me all
collect
that
things that you can
siphonophore?
put away right away.
Oh yeah. Okay. Yeah, I'm done.
Okay, go on.
Do no glass to work. You got plenty.
I got plenty. Yeah.
Small tracking.
Oh, an Octopedema,
And
Coelodendron,
Coelodendron.
all these nice things.
Trachy? Medusa?
Yeah, Trachy Medusa, Octopedema. I'm actually looking for a mature Octopedema because the one we got was immature, but it's new.
I think that's just a kitty now. No, squid.
Squid?
You got a squid?
No,
No.
no, it's not a squid. It's a tomopterid. Stop.
Tomopterus up top?
Oh,
It's an orange tomopterid. This is a target.
You sure it's not a lepidorhynchid? I think it's a lepidorhynchid.
It's
Is
so
it? Or is it-
pretty.
It's just a tomopterid. Karen!
Karen! Karen, where are you?
Come up with the round.
That's right here.
Very bright orange.
It is. We're gonna have to stop collecting for her soon too, because she's getting it too much queued up in the cold room because she's
What am I gonna call in?
too busy.
Sue?
That out? Do you want
Let's try and get a little bit of video and then we're gonna suck it up.
Okay. Yeah, bring it back in a little more.
Really want.
Yeah.
But if you're having too much trouble keeping it controlled, then go right to suck. Your call.
Hmm.
Did we lose it?
Yeah, I think I saw him shoot way back early on.
Poliquita.
See that again.
Poliquita.
Poliqueto.
I don't see
Poliqueta.
it.
Poliqueto.
Poliqueta. Octopedema! Okay, I'm gonna spend the rest of this dive looking for stuff for other people to process.
Any break?
You can go upstairs.
That was a little Kelly Coffin in close.
That's a protist, I reckon. At least the one
I
that
was
came
trying
from
to
far
decide if
away.
it might have been a Dolly Old or Dolly Upsid.
Ooh.
Over to the, yep,
missed it.
have a
What's
look at
that?
that. Already hit it with the vehicle, I guess.
That's a bit of fascinating tail. But there was something down below that was wrapped up
You
around
did
it.
see it, didn't you?
I think we should find a nice. Oh,
Small parade?
is that a desmophis?
No, it's beating like a
it's
rose.
a small crane, yeah. And with the red stomach, it's probably Desmophis hematogasta.
Siphonophore, yeah, Calicopher.
Getting close to see if the somatocyst is going into the mesoglea after running apically. What's the white spot on top? Is it a hyperinaptopod sitting on top of him?
He's right at the edge of the camera of the light field.
Oh, he's just gone right. Oh, there's his tail. That's an oink, isn't it?
Yep.
Maybe he wants the break.
Wash
my hands, which are
He told John to put it away. It was
Karen, we
so-
saw a golden
That's
spot.
right.
Mhm.
Do you
Crazy animal.
want time to work more with your samples, or is sitting in this chair a good break for you?
I need a break. I've been doing samples all day.
Got it. Okay. Well, I'm here to help if you need it.
Yeah.
So,
All right.
It's a halocreatid?
great.
Did,
Yeah, it's a
did John
hyperinaptopod.
come back down yet?
Yeah.
Okay, so he's got the material from Google on it?
Yeah.
All right, good.
It's a medical?
Tracking.
Some great conversation.
A golden slop dress. Oh.
Fish.
Did we collect it?
Show John where the next thing is. Okay, next thing he's gonna process. I'll be back.
Golden Sumatra.
If you see Manu,
Up here.
Yeah.
they'll him there.
I
It's kind of
Yeah, yeah.
thought I saw when we were looking at something in the art, and like something went by, and it was yellow, and it was
Oh.
very furious looking.
Was it moving?
It was not, so it was getting swirled.
I feel like I saw one with maybe a remnant of a net, but like also not sure.
I mean, I have a species
Was he
in
so
Cape Verde, so there is
Something
one,
else. Do you think it's rats?
and it is such a good species.
Sorry.
Nice vintages.
Is it normally on manual or is it on?
Didn't we set the color,
What's that?
didn't we play with the color settings or the mode? You were giving directions on how we set up the camera.
Earlier, it
last-
was Zach said just a matter of more lights.
That one side.
No, that's what Marco did earlier too.
So he-
We were happier.
Yeah, here we go. Yeah, let's see, these, usually we have the lowers off, nose out, down.
Yeah, that, this is a bit better.
Was it cyclotron, the fish?
I think so.
Cyclotron. Dutch too, where there's a lot of words in Dutch that have two completely different meanings. Because there aren't as many words.
CY, ciclotone, CY.
Sim. C de caça, Y, C, L, O, T, H, O, N, E.
Gracias.
Is that a rise area?
Oh, everybody.
Or a doliopsis? So I'm trying to figure out.
I can't tell necessarily without doing the full auxiliary camera correction versus
Holodendrio?
Yeah, it could be small ctenophore as well, lobate.
Alex Eagle asked if I have anything for it, and those have all been good.
There were a lot of what I think are.
Try and get it in a frame.
There's not been any seems to be doing okay.
No, it's too swirly. Okay, move on. We had both the fish and a kitty out there.
What should we do?
And that way. I think it's siphonophore. Might have been that one that went by us a few minutes ago. Well, it's not a siphonophore. Just a bisonet. Okay.
Yeah, this looks like an anemia.
Okay, zoom.
All right, that should be good enough.
Anemia.
Is it, or is it where to go?
The world will never know.
Well, Dougal will review all this stuff.
It does look like pneumonia.
I think it looks like pneumonia.
Pneumonia.
You've seen pneumonia
Around
once.
the ectophores. This deep, they can be
Yeah.
found.
Good look at it.
Yeah. Is it?
Well, if it is pneumonia, we should collect it. You know, it's probably
I thought we've already.
Or maybe this is the OG Bajuga.
All right, so I'm gonna look at data products. That's interesting. Well, it looks like.
Okay,
not that
zoom back
much
and move on.
Indicative of the rest of the ocean, but there is a peak
Pinhole.
at 350, and we have
Okay. Where is it?
range to 700
This one?
meters.
Yeah. Siphonophore.
It's a
Is that
Fathomverse.
part of it on the right?
Huh?
There you go. There's a big one for you, Dougal.
That's a
No, we need-
Then
We need it.
It is a
Yeah, it looks like
Fisonec
Timothy.
tinophore. He'll give you the name.
Fluorescence,
We're,
yeah.
we were
Timothy's
focused on-
Or not a
multi-dentata.
Fisonec, the Cali
Yeah.
Conference. Sorry.
No.
Multi-dentata.
Took picture while it was out.
I do the same thing with
Really?
my kids. I get their names mixed
Yeah,
up when
yeah.
I know better.
And it's so stubby compared to normal multi-dentata.
Oh no, it's good. Leaf is very abundant here.
Not feeding.
Multidentata. The shape, you can see the shape of
The
hydroacid.
somatosis, yeah.
And somatosis, the
Yeah. Looks a little different, but yeah, I think so.
Olá a todos, Portunhol está de volta.
Yeah. Is Fisonec still there as well?
Esse
That's
é um,
good, thank you.
que tem sifonóforo calicóforo.
Is the Fisonec still there on the right, or did he? There it is.
Yeah.
Está lá ainda. Esse nós não identificamos.
Did you not
This
get any
one we
grabs
did
of
not
it?
identify yet.
Is that a
This-
Rhodochovia?
Not
Yeah.
sure.
Oh yes, it's a Rhodochovia.
Oh,
It's a who?
did we have you already captured these guys?
No, we haven't.
So let's collect them or
Fisonec, yeah.
Fisonec.
Zoom down all the way here.
How do
Great.
you spell it?
Are we collecting them?
Can you spell it? Yeah.
R-U-D.
R-
J-A-
R.
R-U-D. J-A. K-O-V-I-A.
K.
We can practice
Radicovia.
letters.
Yeah.
And it's Portuguese.
Radicovia plicata. Never saw this. Plicata?
Rodgico, well, it won't be plicata. That's the only one currently described. But
Okay.
Are we
Should I?
collect them?
Should I?
Do you want him, Dougal? You're
Manco-san
full,
is
right?
working on it. Yeah, I'm full.
Okay,
Maciek
go
is
ahead.
working on it. He's-
Oh,
he can come
Manco-san.
catch it himself.
Ah, I see. Okay. Okay. Nós vimos um calicóforo, um sifonóforo calicóforo com 2 oides apenas e com as bordas
615
fluorescentes,
meters.
uma cor verde muito bonita.
Is that the...
I don't think I've ever seen anywhere that has close
Something like that, probably Keating Ness.
E os calicóforos são
Fish
sifonóforos
like
que não
less
possuem
sinusoidal, much more
flutuador, né? Ele não
Kinda
tem
like
aquela
wavy
bolsa
like
de
that.
gás
Instead of
como as
sinusoidal, like-
caravelas, né? As fragatas portuguesas, né? Ou caravelas portuguesas em português, como elas possuem, né? E nem como e também esses fisonectes possuem o primeiro zooide, né? Que vem à frente da colônia. Nesses grupos de animais é um flutuador, possui gás dentro. Esse calicóforo é um grupo de sifonóforos que não possui pneumatóforo, né? Apenas os nectóforos que são
Yeah, let's see what that
propulsores da colônia. O que temos aqui? Olha, um pedaço de
It's like it might be.
sifonóforo. Pedaços de uma
I'm
colônia.
not sure. I think it's kind of inclined to think it's a house of some type. No,
It's
it's
got
not.
siphons swimming.
Nope, nope.
Yeah.
It's another sight. But
It's a very short
oh,
siphonophore.
it's lost its tail.
Yeah.
It's like 3 or 4 nectophores and a float
Yeah.
on the right hand side.
Mm.
It can grow its tail back again.
Okay.
The budding zone is still
Yeah,
there.
yeah.
All right.
So, esse é
you
um
keep going.
fragmento de um sifonóforo.
Not going to get far without a gas.
Esse, temos o flutuador ali à frente, 3 nectóforos e um pedaço de sifossoma. Então, como a zona de crescimento desse sifonóforo ainda está presente, esse animal vai se regenerar e continuar a crescer.
Faisone?
Faz o nex, ó.
Yes.
I missed it.
Pisone.
Over here, it looks like maybe Solmissus.
Sifozoa.
Is that a frilligana there? Siphozoa.
Oh,
Solmissus.
Solmissus.
Is that a hyprid on its bell? No, it's a reflection.
Ok.
Oh,
And
we
host
could
just in the
zoom
lower
in and
corner.
find out, I guess.
Can we? You don't need to see it, Dougal. You can go on.
I think we should zoom in and check for hybrid.
Clever.
Ah, que imagem linda.
Psychedelic.
Ok. Esse é narco.
Narcomedusa.
Because it has stomach pouches and it does not have otopulpi or lines of nematocysts around the edge of the bell.
Okay.
Ok.
I will
Solmissus.
write
Oh, is it covered in nematocysts? It looks kind of not completely transparent, doesn't it? You know what you're doing.
We're gonna move on after this.
Oh, really? Do you reckon?
Uau.
I think it is covered in nematocysts, isn't it?
Uma medusa agora do gênero Solmissus, uma narcomedusa que tem esse véu que fica pulsando bem bonito e esses tentáculos
I can't see
que,
patches.
na verdade, os tentáculos numa postura normal desse animal, eles ficam
Maybe it's just a fake.
pra cima, né?
Maybe it's sick.
Ela só tá assim porque ela tá nadando, mas essas narcomedusas, geralmente, elas têm os tentáculos direcionados pra cima.
That's pretty good.
These statocysts, the bright dots of
It's
statocysts.
got some brooding pouches.
Brooding.
Where it broods the larvae. They look like
Ah,
eggs,
yeah.
but I think Karen actually wrote
Yeah.
about it.
About the
That it's for
narcomedusa.
the larvae live in them.
Yeah. Because also in Cunina is common to see
Mm-hmm.
the
Little babies.
even larger, not just larvae. You see
Lots
the
of larvae
jellyfish. Yeah,
attached,
the
yeah.
jellyfish attached there.
Okay, thank you very much.
Okay.
None of them have anything in their stomachs. It's just that one who was eating the lobate earlier.
E esses pontinhos brancos na medusa são larvas que essa narcomedusa ela carrega, né? Então, estava comentando aqui com o professor Dougal que algumas espécies de medusas desse grupo, você consegue enxergar as larvas, até medusas pequenas, dentro da medusa mãe, né? Então, tipo de viviparidade, né? A mãe carregando
Probably oik.
a prole,
Go ahead
né,
if you
a sua
want to set
geração.
up and go right into a D-sampler with it.
Oh, que é um
that-
larváceo ali.
Oh, tell me first.
Yep.
Because I'm learning a
Oh, he just
longer...
escaped.
Did he just
escaped.
jump?
Yeah. I saw him in the camera that's pointing right.
He's running.
He's running.
If you're lying, we're going to
Well, I
No,
log
it's
you.
here, it's here. It's- yeah, yeah,
There
it's
he is.
there.
Yeah. If you want to go
Must have
after
been the
that, we
other
go...
one.
Sorry to say, but
You can't trust a word
Fritillaria.
says in this room.
I'm going to try a different strategy. When we see one, let's go right to collecting. So we just...
Where's that one?
Less maneuvering.
In the Midwater zone.
Yeah.
Eat or be
No, I
Fritillaria or
Yeah, Oikoplura.
Oikopleuridea?
He was up a little high. That probably put in the wash zone.
Everyone's out to eat
So
everyone
what?
else.
Whether you're in the boat or in the
When you're in the boat or under the-
You want.
Oikopleura, no species. Oikopleura, only genus.
And there's a Calicophora. And was that a fire snake as well? Or Falkor, maybe? Like a Medusa, maybe. No. Bottom right there.
Medusa, bottom right, no, bottom.
Okay, great.
Very descriptive.
Everyone, midnight meal is meat. According to, oh my God, rib night again? It's crazy to eat ribs in the middle of the night. Yeah. Dinner was really good though. Something. These days and for the past 200 years.
Yeah, right. No road. Famous one.
Nope, that's an old one. That's collapsed.
Oh, the nastiest. Oh.
I'm gonna switch seats.
You wanna take control here?
Yeah. Thank you very much. Okay. Do you want to do it on the PC? Actually, doing the tape? Yeah, doing the download as well.
So this is...
Oh, it's a So Mrs. Why is no one excited?
Yay!
Yeah. Woohoo! It's only 11:30, guys.
This is probably the same one, yeah.
Yeah.
The rate we're going up and down.
Slightly back and then curl them at the end.
Yeah. He's got that twirly thing happening here.
Yeah.
Oh, wonder why that happens.
Okay. Do we need more of this?
I think we're okay.
All right. Move along.
So Mrs. Not gonna use it.
Wait, have we actually done so Mrs. We have some, actually, we have some iris data with some misses, but.
With
One
some
iris
misses.
stuff is just, how are we gonna, process it? Need some people who specialize in image analysis.
Good luck finding that.
Look around the room and you're like, uh-oh. Uh-oh.
If not here.
Ooh. Very rare, very beautiful.
Might
Is that
be
a Doliolid?
Doliopsis.
This one was Peter.
Doliopsis. No, I think it's
No, it's
Harbisoni, isn't
something
it?
different.
Paradoliopsis, no? Something else? Yeehaw. It was a Doliolid for sure, but which one, we're not quite sure yet.
I think you might be right. I think it might be Paradoliopsis.
Paradoliopsis, yeah?
Okay, zoom.
Yeah, because the 1/8, the buccal siphon's fairly open.
Let's see.
Yeah.
Can I close it?
Yeah.
Oh, I just do this.
We're going to put this in a D-sampler ultimately. It's up to you if you want to get some better footage first or we'll go into collecting mode.
Put your porch out there.
For inside?
Yeah.
John?
Or the
The IP. Yeah, I don't know about Windows. I'm just kind of guessing.
Put the light back on. They're all on already.
And you'd have
Okay,
to
follow
put
it
in
with
like how are you
the
going to talk
side
to
cam,
it, so
yeah.
this soundbar.
It's probably about 2 centimeters in size.
See what happens.
Zoom in all of it.
It needs a new app.
Down.
No, normally it just works.
Close.
In.
Oh, it's in. What was that? I missed the entire thing.
Good job. The one tab's not tight, but as long as the bottom's good, we might be okay.
Oh, they can close that with the manipulator.
T4.
Was that the paradoliopsis?
That was the Paradoliopsis, yes.
I mean, I'll do it.
And that's in one?
Is that? That's D1.
Oh boy. I'm a little intimidated by that one.
Someone's begging for Sylvina.
Silvina is
So why didn't we do this for a light balance then? Just move the porch out.
1912 or 1913?
Zoom in.
This one is the 1913.
Yeah, I think 913 is the right cursor. Right?
Oh, I'm sorry.
13. I think the last one was 12. No? Yeah, 913.
It's 7:30 over there.
You can come to my house and hang out. You can come to my house and hang out on the hammock.
An hour ago.
Perfect, thank you.
Just want to get out and see.
This has got a web browser on it somewhere, I guess. Does it have internet?
It's alright, you don't have it. Sometimes it's better if you forgot.
I feel that she's a little that way a little bit now. I feel down there.
Hey, you want to go straight down? See that particle right in the center?
How to access a network using
Shouldn't need to go far. There we go.
Excellent work, so we don't even need to be here.
Colubenema.
It is a Medusa.
Colubenema sericium.
Oh, this happened, yeah. My university likes blocking everything. What do you guys
He's
trying to do?
on in a hurry.
We want to use Samba to connect to my NAS, so we can copy the files we downloaded from the
So I don't think this would actually allow me to do it because I don't have admin rights on this laptop without being at the university.
Are you able to connect?
Dougal, we've got this or not?
Sericium. Oh, I don't think we did actually.
What?
I don't think we've caught it here. No, we haven't. This
Okay.
would be a fast process.
Yeah, I mean, you're not going to need to spend a lot of time IDing it. You're pretty
Yeah.
confident on the ID.
And we don't, do we need any scans?
You can if you want. It'd be nice. Yeah, because I guess we can cut it in half for the genetics too if we lose the tentacles. So we can do that. And isotopes too. Yeah. Yeah, we can scan if you want.
What size do you think it is?
Is there a way to map to it? Just from the PC, like to a network drive?
You don't normally join a network.
So are we scanning?
Oh, it's a good advice, actually.
We're going to scan it then?
Yeah, if we get a quick scan, if we start with Iris on the left.
I've been at the university. I just got this also.
Sure, yep, still do
Do
it.
you know, like, assuming it could already do it, and you just haven't done it, would you know how to do it if? Because I'm not a Windows person.
No, so do you have a server hub like this?
No, you could just,
Maybe.
I had the mids off and just in and off the uppers one.
Yeah, I've got an IP address.
And I was trying to find for the PID one.
But I mean, just.
But this one you can leave all the lights on right
169.254.
now. Could you turn the
100.
dark
I
camera,
don't think we need 60.
the side camera?
And then browse. We have to look for the folder.
Otherwise, we can also just try. Okay, wait, no. Let me just try. You guys try typing it into the
I tried typing it into the.
It didn't work?
Didn't work. Mine does, but I have a
That's
Windows
great
computer
framing.
where I must have joined once before and it's remembered it.
Is it okay if we disconnect this?
It's the perfect size for this fish.
Can I take it up? Yeah. It's okay if I pull it to the right.
I can play around with it for a while.
Okay. No, I haven't connected to the Wi-Fi here, so I can set that up unless you have a voucher.
It's starting to come in.
Didn't think I was using
And a
the
little
laptop.
far.
Yeah, if you have a Wi-Fi voucher, connect to the network and then go from there.
Far. It's looking really good in the field of view. Just a
Do you
little
have
bit
any
far.
external hard drive at all?
How much
Is
that
this
has that?
only? It's like around 500.
Nice, nice, nice. Okay, we can move on to DPID.
Yeah, if we had that, we'd be copying directly to that. Russ might have an SSD that could be formatted to do 500 gig. So we'd have to copy from that to that and then use that for the other one to go to
Leave
that.
the uppers on, between
Let me try this.
no, leave the muds on, turn mids on, lowers
Also
off.
takes time.
500 gigs, yeah.
Good Lord.
I mean,
I think we leave the center cap, turn
it's
off
like
the
one
lower
of those,
cap.
solid, yeah, some of those will do it.
SSDs, yeah.
Yep, I'll be ready with the others.
We could look at the waterproof Windows machine and see how that is connecting to the NAS. And see if we can do the same thing with this one.
We could. The only worry is that it locks you from doing a lot of stuff.
Yeah, no, this one is Thank you for setting the very happy.
Okay. Stairs are high. A little high.
Oh.
Great. A little high. Keep it coming, keep it coming. Nice.
That looked
Okay.
good. Yeah.
One more time, reverse.
Fill up my experimental thing.
We'll take that, thank you.
Okay. Suck them up.
I've learned not to stop recording.
Good plan.
All right, thank you.
Are we collecting?
Are we collecting? Yes.
Yep.
Yeah.
At some point, I'd like Suction?
Suction.
And it didn't drop its tentacles.
Any lights back on?
He's just gonna swim in on his own.
Yeesh.
Oh, almost.
I was gonna say, if he like literally swam in there.
Oh, Fran.
I got it.
Columbinema sericium. S-E-R-I-C-E. And we caught a Columbinema apicantum earlier today.
Zooming all of us on the thing is close to the.
Yeah, I'm surprised.
So many of these. Don't stop recording.
Oh, I bet Kakani knows how to use Windows. You're a Windows user? Or you use Mac?
I use Mac primarily. Embarrassingly, I haven't used Windows in a really long time, and EOS asked me to restart that computer, and I could not find where the restart button was.
Yeah. Yeah, I mean,
I was like, this is embarrassing. Where is it?
we went up through Windows, you know, up to 10 or whatever, and I was using
Got some
it
of the
in
tentacles.
XP emulation mode. I just didn't know where everything was. So I went to 11. I'm like, I got no idea.
I was so embarrassed because it was like, see, he asked me to do that one.
Oh, he's in! Hey! Woo!
Hey.
Turn off
Okay,
the pump.
same thing. Shut the pump off for a second. Let him, and then give it a click again.
Me?
No, pilot. Okay, give it a kick again.
Which number is the sample?
There it is. Beautiful.
5, did you say?
Yeah, 5.
5.
Yes, five.
Now I haven't seen one.
Yeah.
It's in a good spot. Nice. Yeah. All right, you guys. Number 6 is the next one. Yeah. Broken.
Not now, no.
House.
Where?
Right there. You go right into collecting mode.
There's one higher.
I guess it'd
Either
be
one.
SMD.
Looks like a team of 4.
That's not a house. I don't think that one's a house. I think that's a jelly
Trying to search
up top. So we want to go for the house. It was just. Where did it done? There it is. Okay, it's gone. So let's take a look at what the jelly was.
We're getting better at this.
You are.
It's like almost like a burroid. No, it's a.
It's not hard enough.
We just get some hortensid from the end.
What is that
This Thalassocalis.
again?
Yeah.
You need a scan of this, I can tell.
Yeah,
When it's rolled up,
We could use a better scan.
because that way it won't move if it's
Yeah.
rolled up.
Could we do one of this really quick?
Yeah.
Our scans haven't been great because
What,
they get sloshed around. DPIV, the
Okay.
laser. But if we kind of deform it, we'll probably not want to.
What are you for, trying to find?
Yep. Scan it.
Put it down as Lobata. It's actually other, but it's close enough.
Loba. So I'm gonna zoom in a little bit.
It's not working.
It's not working.
Well, I thought it would come up automatically, and it's not. So let's just play around and see whether it's possible in Windows to just type something like this in and have it open something up. This is going to open up the home page of the NAS, which is not what we want to do. Because if you access it through a browser, it'll go through the port that goes to the NASA's web server. So that's not what we want to do.
It was a little fast, but we could take
This
that.
is the only thing that we need. Right? So
Whoa.
in Linux,
That's
there's a
weird.
little.
It's rolled up.
Yeah, it does
really
that.
is like
Yeah.
completely rolled up.
It rolls itself up, especially when it's caught something, like it's eating a Balanopsis or something like that. It'll roll itself up and have its prey inside.
Yeah, let me zoom in.
So you got 2 good scans on it?
Let's do one more.
Does it have a hypergrid up on the corner there?
Oh.
Beautiful.
Right, one more backing out.
Okay, that was quick.
We will take that. Thank you. Thank you.
All right. Let's go hunting for some
I
houses.
know. We need to find harder targets.
This is amazing.
Harder targets.
We're still after a larvae.
Larvation is going to be.
Search network. Maybe they come up here.
It's the one that eludes us. Just typing it everywhere you can.
Yeah, you will.
I missed it. Over here.
I don't normally copy to a NAS using a Windows machine. I normally take home
Hosts.
a external
Nope.
hard drive, 5
Joe.
terabytes or so, and plug it into my
Yes,
Mac and copy
host.
over. But I've done enough engineering troubleshooting that it's like, try this.
He's
try
still
that.
there.
Did you do
About to be hit
that?
by the wash though.
It's hate that it's-
Give it about 4
We
to 5
believe that's a
seconds. See if it-
Thalassocalisi or
Yeah, we could capture it. Yeah.
That is an oikoplura of some sort.
That is an oikoplura.
No, it's a lavation.
Do you you want to collect this?
Yep, we're going to take it in the other D sampler.
Okay, do your practice run with that number.
It's already starting to get mixed up a little bit. It's left, okay?
Do you zoom out on the?
No.
Okay, coming in. Oh, here you go. So it's coming up automatically there.
Yeah.
So in a, not a browser, in the OS search bar, 2 slashes and then the IP. No, it says it can't access. Oh, that's because we're not plugged in. Right?
Close. You got it. Good job.
You got it?
Is the animal in there?
Yeah,
Sample
it
in
is still
D1.
attached to the house. Let's zoom in for a sec. Wait for it to come to rest. Oikoplura.
Looked like it was.
I'm sure it went in. I just don't see it yet circulating past the window. There it goes. I just saw something swirl around.
Cannot access. Ah, because it's probably, the IP address is probably still set to the download of the shadow graph folder.
I'm
or
pretty
something
certain
like
it's
that.
there.
That's a possibility, right? So go to control panel, because we haven't reset the NASA's thing yet. Ethernet, properties, TCP. Whoops, hit the wrong thing. Let's do this one. And yeah, it's still on the shadow graph we're not going to be using this.
know, Russ, are
Are we
you
using this to copy?
confident
Did the
that-
shadow go fast?
I am certain
You
it
just
went in.
You just
Alright, so
downloaded,
Russ
right?
is
So this
confident.
is useful that we're doing.
Could have
this.
could have gone down the slide and we thought it went in.
It's always a possibility, but I'm just surprised.
I was confident it
Okay,
went in.
so this should now be on the network,
Alright, so you got two people that are
which
confident.
means if I
Right.
press
we-
that,
Or two people that are wrong.
Yeah.
so it's a slash
did
slash
you
and then the IP address.
Actually, we
Need
could
her
be both.
computer.
I mean,
Well,
who
I guess
knows?
we can copy first, right?
We'll find out later.
Could you poach out email?
Gotta go look at it.
And it was SO911, I guess.
I'm surprised we don't see the house, though. It did go in with the house.
It should be written on.
No, this is the best because I came back in with the camera. He gave us a little.
I mean, it could
Yeah,
still be right up
right,
against the door.
We're on 913 now.
There it is.
And
Phew.
that means
Was that inside? No, that was
Yeah.
behind
No,
it.
that's right.
So it goes to go into
No,
there.
that was outside.
That is,
From
that is a different kind of animal.
download tickets.
Yeah.
It's one of the amphipods.
It's a team
Desktop.
I mean, the sites so far.
Maybe SH1.
I am really surprised we don't see it. Normally, at this point, we can see it if it's in there.
Yeah, I think it was a false capture there. We thought we got it, but we didn't. Shake it again, fortunately.
Well, there's not a lot of more shaking we can do. I mean, the animals.
I mean, shaking does help.
Other way?
Or, well, I go back out.
Other way, yeah.
So that's how you do it on this computer.
Let's see if we can do it on this one.
Should have seen it by now.
Yeah.
I think it was false capture.
Well, if we get another one, we can always open in the process of
Yeah.
a capture.
Keep-
We won't open it again until we actually have something to replace it with.
I did set for number 4 for the D1 I set those to 0. So D2 I'll leave just in case.
The best way to capture dolly opposite chain is when it's inside a Solmis's gut, because then they're really
Oh.
easy to catch.
That is a wonderful story. Okay, let's keep searching.
What do you think that facial expression is?
I miss your mommy.
I don't think so. I think
I'm hungry.
Oh yeah, I'm hungry too. Oh, we missed dinner.
Slowly set me.
What did we do?
I think we just missed the midnight snack, right?
No, it's
Oh no, it's still happening. All right.
we have half an hour.
Okay. Another Thalassocalis.
Look back at this and like she was such a baby.
Oikopura.
Yeah.
Is that just one?
I think it's the same one we were looking at before.
Oh. Does it have something in it? No, it's just bunched up the.
That's no alarm page.
No, it's not a larvacean.
Ah, what is this one?
Great squigglies in there.
What is this one?
It's either.
Ctenophore.
Oh.
It's definitely a little big ctenophore.
Oh,
It's got.
Look at those canals.
no bata?
I think it's the
Try lower
Thalassocalis.
zones, yeah.
It's almost a lobata. Soon it will be. Right? Right?
Right.
Working Google.
It's got no oracles.
Not definitely a lobata.
No.
There's no like gonad kind of area like Thalassocalis normally has, but the gut's right, and the tentacle bulge being that
This one
far
is moving,
apart
so
on
it
either
should
side
get
of the
some
mouth
discount.
is also a Thalassocalis. It's got to be Thalassocalis. Maybe it's just reproduced and it's got nothing left.
Yeah, I don't see the diverticula.
Spent.
That happens.
It's just lying there now.
Otherwise, it looks right. the lasso, like in C, THALY.
Species?
Yeah.
So, no, wait, Val,
It's very old.
THALL.
Must be old. Yeah, you should scan it.
It'd be good for the Irish Art Gallery.
Oh, yeah, can we do, we don't want to collect, but if we could do an iris followed by a PIV scan. Same as before, we just want to be careful about too much vehicle wash, right? Because it's in a really nice pose right now.
Okay.
Oh, did you see it retract like the sphincter muscle, the ring muscle?
Yeah.
Oh, did I?
You went. You went what?
No.
It just went you and tightened up. Choop.
If only that happened. Whoa, almost. Back in the center.
Distracted by Doodle's story.
That's how the catches krill and stuff. Krill goes in there and whoop, sphincter muscle just closes and it's stuck inside and it puts its tentacles down and catches them. 35% complete.
That's too bad. All right. Thank you very much. We'll just do that because its shape is really different now. Thank you. All right, next animal. We're supposed to pull at 2 a.m.
2 a.m. on deck.
Or
So this is,
Is that an apolemia?
no, ice neck. What is it?
Looks kind of weird.
today is Friday.
It's just a loose tentacle
Oh, maybe, yeah.
thrown off by colopodema or something?
Yeah, it does look like a colobonema tentacle. How's that? I reckon you're right. I reckon it's a.
Schedule again. Every time.
Oh no, there's a gastrozoid. It's one of
I'm ready
those
for that.
siphonophore cords. You know, look at the gastrozoid.
It's just a loose
No,
tentacle
oh,
or
it's wrapped
something.
around itself.
Oh no, Friday is still happening.
Yeah, you might be right. It might be a colobonema tentacle without the medusa. That white distal end, very long,
We're just shifting everything back.
flexible.
And then who knows what after that.
Colobonema, Tracheomedusa.
Ah.
We think.
It looks like it's okay.
I mean, it looks like a colobonema tentacle.
Okay, move on.
Moving on. Thank you.
Top left.
Little medusa there.
Oh yeah, Octopodema.
And friend. Got something stuck in the tentacles. Okay, keep going.
Can't see much in the way of gonads.
No.
No, Tracheomedusa. Octopodema. Octopodema. Ooh.
We've seen this before.
It looks like a colidarian.
It does
but
look more like a colony, doesn't it?
Yeah, that'd be cool in a scab. It's a donut. Whoa. It was pretty small.
It was.
It's a ring. Why is it joined together like that? Colidaria colony. A rhizarian
rhizarian.
colony, yeah. Because I don't think you can lay eggs like that. It'd be really hard, like physically. So it must be a colored area.
Oh, that's too bad. That would have been really interesting.
Oh, it's a classified. Either a Coniferes or a Clausophis. It's looking like a Coniferes.
Little vice connect off to the other side.
It's Coniferes multidentata.
Okay.
Oh, capture button, capture. Nice. They tend to hang at that angle. It's pretty indicative that it's a classified, and they're big. Is that a Desmophis hematogasta I see before me?
What's below it? Right there.
It's got red gastrozoids. And
It's
a
is that a house below
siphonophore. It's a house below it, yeah.
We want to look at the house.
Okay.
There's a larvation.
I think it's Desmophis.
That's a larvation house.
It includes the animal. It's in there.
Yeah.
Oh, no, it's not.
No,
I can't see it, yeah.
can't tell.
That animal is still there.
Oh, yeah. There's his tail.
Huh? In the house. For the moment. Looks like the house is shearing right now.
So you can shift it.
Inside the light, right?
Maybe just let me know.
That will blow him up.
If there's mashed potatoes, I might. No.
Okay, let's take another look at that D sampler and see if we can see anything in it now. In case he's just hidden.
Question mark there. So if you guys see one,
Tree
that's
sampler.
like ours. Right, so it's done. Looks this way, so you can still look at those out.
This?
So what we've been doing is when we like zoom way in.
Nope.
15,000.
Still nothing.
Yeah, I think
Does that include it as well?
I think we missed it.
How much storage do you?
I just wanted to check before we consider throwing it away.
Good question.
So we're
It's not full yet, I assume.
Yeah.
We have to go into the
Alright.
At the very least, 4 terabytes.
Okay, that's good.
The one you captured today was a much smaller species than we've been trying to
Yes,
capture.
we were all talking about snacking. Is that a coelodendron?
Just leave it for a while.
All right, let's see if we can find a house to replace
I
that.
think
with.
so.
You got the guys to do it.
Just
Yep.
unplug it?
Okay.
Yeah.
Out of here.
Say that again?
So you leave it till at least after you eat.
What is up there for food?
Ribs.
Oh, we didn't know.
I just realized it's midnight and I was like,
Who's that?
Okay, Ben.
No, you just don't feel like
another
working,
house.
and it's a.
Yeah,
Look at that larvation.
another larvacean house. We go after that in the D sampler.
Let me check. I might have
We're gonna throw away whatever's in D. Oh, he's already bailed.
I might have like 2 terabytes or something.
Alright, I think
I'll
after
double
2
check.
a.m. recovery contact. Known as
It's got, you know how universities like to install security.
I think so. You can
It's not
you
letting
can
us
zoom in
install
on it, but I'm
the,
pretty sure I
NAS
saw him
code
leave.
Saving our data to.
that's why you run your computer on Linux and they can't, they don't have any
Yeah.
security software for it.
Yeah. I
honestly.
That's what my other computer is, but that's not here.
I've got one, but apart from this, I've got one Windows computer in my lab, but it's running the Linux subsystem for almost everything. Thank you.
Thank you.
Yeah, I mean, are we planning at some point to have both cameras at the same time? Or?
Yes, if we can ever get this download and charging thing working properly. Like one dive on, one dive off, with both running is for the project. That's what we want to do.
I want to see if he's still there. He might still be there.
I'll see if I have my XR hard drive.
Yeah.
Okay. Thank you.
But yeah, for the one that's running, I think we can use this, right? Transfer stuff.
See the center band?
It's a stereo.
The animal went up that way.
Maybe, yeah.
Oh, you got it in that
can
camera.
try.
Okay,
I'm
I
not
couldn't
sure
see
if
that.
it's
I think he might still be there. I thought I saw him jump.
got enough space on it. But if it does, then yes. As long as we delete, do we delete the shadow cam stuff yet? Yeah. Then maybe we'll be okay.
Try to find them before I'll watch it. So, it's okay. See that one?
If we run that software to do the download, and then manually delete the files.
If we determine he's in the...
Take a nap and waking up. And we'll come back. Two?
Oh, was that a Nemodian on the left?
Do you want to take a nap? Do you want to...
Well, the house is a structured construction. A
Yeah, I
mucus
wouldn't mind
blob
seeing
may
what's
be a
there.
contracted house, or it may have actually originated from something
Some
other
fruit
than a
or
house.
something.
Better current, yeah.
What are those? Is it too late? Just curious.
We have a XR hard drive.
They look too symmetrical.
Yeah, we do. What size do you need?
Around 500 gigs.
Oh, yeah. But do you need it for a very long time?
No,
or-
just...
Put it in center.
Yeah, we have a bunch of like 1 or 2 terabyte.
Zoom in a little tighter.
Yeah, just zoom in. It just, they
I might
look
have one
exactly the same.
In case.
I'm trying to figure out what they are.
I'll have to see what we have on
Closer.
Yeah. It's a pair of Rhizaria. You can see some of the structure. Is it, are they Coelodendria? Just with light tentacles? They keep creeping in,
Put on.
or zooming in, whichever.
I'll creep in. I'll creep in onto.
Ignore the fish, it's just there to distract you.
Do we know what kind of Rhizaria is that?
In.
Not yet.
In.
Philodendron. Okay. I don't think they're on the wish list for this
Yeah,
dive.
yep.
Are they?
I thought, no,
Yeah.
Oh, then trick it
Yep.
update?
yeah, I think so.
It's pretty close. Yep.
So they are on the wish list?
I'm not
Are they, or?
I have no
It's
idea.
my new one.
Oh.
Shall we collect them then?
Oh yeah, we can go.
Tuck them up? Or are they a D-sampler wish list? Looks like they're caught on something.
Still there. So.
Down. Gone. Yeah. All right, let's keep looking for a house or for doliolets, those little spheres.
Can you open the iris in the silver?
Hmm?
There you go. That is. It's been on the sip.
Fish way out there. It's like the story.
Forward.
House down there. I don't think I'm gonna look at it though. Center. It's more like a jelly or a siphonophore, but we'll see.
Again.
Well, it could be a house.
What's that?
It is. It's a house. And I think the white dots.
Let's leave it there for now. I'm gonna get closer.
Still there.
All over the camera. Try this sometime.
Okay.
A little tighter. 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 Too small. That time?
Outside the.
He went outside. And he will have probably picked up enough turbulence in that to be. I'm not sure he's still there. He's right there. And that should be enough to make him leave. Yeah, he's gone. Okay, we've taken a couple of tries of those. Let's see if we can't find a dolly opposite instead. They tend to be a little bit more forgiving. See what that is. Looks like it's just a Feiseneck. Or is it a fish hanging out there?
Zoom in on that one.
Yeah. Doesn't feel like it's getting closer. It's wonder if something we're chasing.
It's moving away. Speed up a little bit.
Is it?
Can you zoom in too from here? Tighten up a little bit.
Oh, it's a siphonophore. It's one of his. Okay.
What kind of siphonophore?
It was a calicopter.
Forward.
There you go.
I think that was just a mucus string, not an animal. Something coming up on the left.
We'll put him center T and then zoom in just so Russ can identify him.
Looks like a halocreatid. Still not, yeah, still not quite close enough to do a full ID. It looks pointy.
Close the iris a little bit. Tighten up a little bit. Zoom.
Yeah, with the helic out it. Okay, move along. Let's try and get a little bit more up velocity and see if we can find some water that has something more or different.
Coming up.
Close. Yeah, a little bit more forward velocity, so you feel like the particles are coming toward you. So the main camera is kind of in a little bit of an eddy, right? The front of the vehicle's up ahead of it. Fish. That is.
Center.
Close. You want to give it a try?
Sure. Go ahead and follow it. Easy.
Still in the house. It's a Fritillaria.
It's in silence. It's about, isn't it?
The animal's still there. Go for it.
You ready?
House.
You ready? Nope.
Still with it.
Where is it?
He's gone. All right. Keep going. see what we got going over there. That was another house. Too late. It's been, and another Kelly coffin.
Russ, do you want to have a?
Yeah, I was gonna have a snack.
There's still stuff out there. So. You got to hurry. We've only got.
Okay, thank you.
Very well. Catch oaks. Libations.
Yeah. So stuff for other people.
Yep. Libations and rosarians.
Yep.
All the way down. Hello. Guten Morgen. No? But we can look at jellies on the way, can't we? Yes, please. Looks like a lobate siphonophore. Looks a lot like a thalassocalus. There's a rhizaria off to the left as well. Yep, that's a thalassocalus. Thank you very much. Onwards, yes please. Someone's asking about the spelling of Coelodendria. Oh, to the left. Oh, on the left, over here. It's a tomopterid. Curled itself up. Do we have many suction samplers left?
I had a sample.
Looks like 6, 7, 8 is free according to this. Yep, have a bit of a look and then suck him up, I guess. That's thalassocalus, the guy we're just looking at.
Keep your pointer at him.
Yes, please.
What is this one?
It's a tomopterus, polychaete worm. Oh, I lost him. It's over here somewhere. Might be up here somewhere. There he is.
Want a video of it first, right?
Yeah, if we can, a zoom in would be great.
Please.
See if there's anything in its coelom. Sometimes they have baby jellyfish that are parasitic and live inside their body cavity. It's curling up again. It's just sitting there, slowly sinking.
He's tired.
Yeah,
Honestly, yeah.
chilling out.
Pick him up.
Yep, I think we're cool to suck him up.
Second.
All partially curled. As soon as he feels that water, I bet he'll be like, meow.
I mean, they're
Beautiful.
About the right? Got kind of a view on this, but it's not perfect.
Yeah, he needs a little bit closer.
Good. Option a little bit for you.
He's on the other side of the fan. It's him. Oh, that was close.
Still.
Oh, look, he's in front and down. Yeah, down here. There he is.
Back with view on the side. Got it.
Is this him, very hard to see, but he's out here somewhere. Forward? I just lost him. Yeah, you gotta go forward. There you go. Somewhere here, maybe. Is that him there? No, it's a feet. Push forward in that direction. Big different guy. It's just almost this. No, it's not him, is it? Somewhere out this way. Try that. No, it's not that. I lost him. Yeah, okay. All right, onwards we go. Okay.
What's the difference between security and
A little Halascara conager just came right up next to the slope gun. Quite a lot of copepods around. Some rosarians. Drive forward, and what depth are we? 537, a little bit up maybe as well. Keep an eye out for some more libation houses. It's a Keatignath. Calicophora and siphonophore, little diphyid.
Yeah, we could try a deep piv, I think, if we do find another Thermopterid. I think Karen's got a lot of samples she's working on already. Might ask her how she feels about it. Here we go, there's something. Maybe lobate. Go in for a bit of a look. That color, rainbow colors, makes it look like a ctenophore. Yep, certainly is. Oh, that's the lobate ctenophore with no oracles that we've been calling Lyria. Or Kappa Lyria in Japanese.
What did you say?
It's a lobata. Strictly it's not, but yes it is. Going for a bit of a close zoom before heading off. That's nice. Go through maybe.
Roboto.
That's a nice one. Oh, there he goes. He's quite small. Not sure how he'd go in a deep piv.
Keep going?
Yep. Do you want to try and scan it or not?
I don't know what size it is.
Yeah.
Quite small.
Is it too small?
Pretty small, I'd say.
That might be hard.
Okay.
Okay, keep going then.
Climbing slowly.
Yep, keep climbing slowly. Would be great. Oh, in the middle there? What do we have here? Is that a... What is that?
I'm bringing the middle to put them in. Yep.
Oh, that was the fishing posture for a Coniphys siphonophore. Go in and see if it's Coniphys or Clausophys. Here.
Closing in. Moving back. It's moving. Moving forward. Slowly moving forward.
I lost him, but that's in there. No, I've lost him. Somewhere down there. Maybe there. Maybe that's him. Possibly that white spot. But possibly not. Well, maybe. Maybe. Oh, it is. Yes. There you go. Nice.
I have emergency Spanish.
Go close. We're not going to collect it, so don't be shy about getting close.
Yeah, go ahead and zoom in.
It's a conifes. You can tell because the ridges are splitting. The lateral ridge is split near the apex. It doesn't seem to have a somatocys perpendicular to the bell, so that's Conifes moseri.
Say that again?
CHUNIP. It's a Chalcophorum, yeah. Chalcophorum siphonophore. Yep, yes, please. And CHUNIPHYES. Moserae. MOSERAE. When I spell that out, should I be like IE?
You know what? Whatever you feel more comfortable.
Whatever I feel comfortable.
Yeah.
My wife is German.
AE.
Well, she's Bavarian, but there's no Bavarian
Still cruising along, looking for libations.
That's fantastic. That's fantastic.
You speak any?
Very little Spanish. I
helpful.
can.
Yes, question in the chat about the spelling of Coelodendria. In Spanish, it would be Coelodendria. 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 It's a Phaeodarian, Rhizarian. In the middle there. Is it maybe another Thalassicalis, perhaps? Maybe do a zoom and find out. Looks like there's a little crustacean crawling over its surface. Yep, that's a Thalassicalis. Is that an amphipod sitting on him, or? Looks like there's something there. We can zoom in and take a look.
What is that, Dougal?
It's a Thalassicalis.
Thalassicalis.
Thalasso. I think that's Greek for ocean. And calis, kind of like chalice. It's a cup. So it's an ocean cup. Yeah, I'm wondering what that is. Take a zoom and see what we can see. What? What is that? Doesn't look to be a hybrid after all, but I don't know what it is. Yep. Film them as we pass by.
Yeah, sea log, taxonomy.
Surprised there are no orcapturids around. Okay, one more zoom and we can continue on, I think. It's weird. Okay. Yep, one final zoom before we go, before you lose him.
Pretty big.
Pretty big, pretty big. Okay, thank you. Krill donating around there.
Forward.
Yep, going forward. Yeah. There's a lot of slightly damaged animals that have been chomped on but have grown themselves back again in today's dive. We saw an Asian Eura earlier on that was a bit like that. That Thalassaculus there. There's a cyclophonia top right. And Salps. A little bit more forward motion, please. Try and get some big things. Rise area. There's a house. Take a little look at that.
Put him in the middle.
Animal still there.
Forward.
Nice side view.
Go ahead and tighten up. Zoom a bit.
Some salps, some krill, other animals that are around. Looks like the animal's still home. Been nice and close. Do you think you'll go in at D sample is full now?
No, there's one available.
There's one available. Let's see if we can try and get it.
Okay, we're gonna try right now.
Ooh, what was that? Scaloides, hybrid amphipods swimming around there. Can't say I've ever eaten a siphonophore. I've chomped on a pyrosome. That was not a pleasant experience. I've eaten krill, salps, jellyfish salad. Yeah, definitely.
Ready? That's good. I like that. It's just outside.
Alrighty. Past them? Yep, I think we've gone past. Let's go look for another one. What cool animals can we find?
My bet is the next animal is at least 5 times as long as it is wide.
Not much happening down here, is there? Maybe we should go. Oh, cyclophone. That's either. That looks like a calicophora and a praying tail. Up there. Oh, straight ahead. Here, there's a medusa. Looks like a Halicreus minimum, maybe? Yes, that's a Halicreus. Keep on going.
Well,
Yep. Moving along, what can we find? Let me see. It's almost 1 o'clock in the morning here, local time in Brazil. We are doing a midwater dive here, searching for animals that we have not yet sampled. We're trying to make a formal catalog of all of the species that are living around here and providing DNA sequences so people can deal with sequences when they're surveying and know that taxonomists have vetted them and made sure that what it says the sequence is what it actually is. So we have a group of taxonomists on board who are involved in that. We're also doing technology work. We have a laser scanning system that takes optical slices through transparent animals and catches them on a high resolution video camera. So if we find any largish, mostly transparent objects floating here in the midwater, we plan to scan them with this deep PIV unit. It's a halicratid there. Rhizarian. Oops, that's a doliolid, is it? Yes, that's a doliolid. Let's take a look at those guys. They're pretty small, I think. Yeah. Going up.
Yeah.
Okay. Alrighty. Let's slowly head up to 450 meters.
Yeah.
An oikoplurid there?
Still there?
Yep. There he is. Undo one zoom before we try and put him in the D-sampler. Rise area in as well. Oh, is that a nobly? It's a nobly.
This is an aquapleurid.
This is an oikoplurid, yeah.
Yeah. Yeah.
And there's also a praying calicofrem in the frame. And a hybrid arthropod.
Keep your eye on him, Google, please.
Looks like it was a Liliopsis or a Desmophis, the praying. Trying to slowly move towards this tiny little fragile organism.
Tighten the picture a little bit up. Yeah, that's it. Broke this suction set. It's really hard. Zoom in a little bit. Zoom. Yes, just ahead. Just Roger. That's coming in the picture. Now it's above this.
Maybe.
Still in the circle. I think he's outside, isn't he?
He's just to the right of the D sampler on the sit.
You go in? He went in. I think he went in.
Goes once. Let's take, oh, maybe. It looked like there was something in there. Let's zoom into the D sampler and see if we can see him swimming around. I thought I saw him like floating.
Might have gone through.
Where is he?
I
can't see him, but he's most likely like somewhere that we can't see here. Colodaria.
Do you want to open that thing up?
I'll leave it. Well, leave it closed for now before we try we may try sampling again with it. We'll zoom in and see whether he's floated into sight then.
That was the larvae.
Yeah. That's D2, right?
on this pool.
Right. Oh, actually right here, shouldn't I? 2.
Forward.
Okay.
Coming up.
Very hard not to stop on jellies. There's so many of them around.
Oh, yeah.
Got about 15 minutes left. Let's start.
All right. We've still got 3 canisters to fill. What's that in the middle there?
Zoom forward. I'm heading that way where those fingers. There you are. Somewhere.
Another little oikoplura. Is he home? See if the animal's in there. I cannot see it. The house still looks pretty inflated. I can't see him in there. No, I think it might be discarded.
Done?
Yep. Okay, let's go find another one.
How do they produce the houses here?
The houses are actually made of, to my knowledge, made of animal cellulose. So similar to what plants use to make wood. And they produce these nets, which they sit under their chin, and then they inflate. Over here? Oh no, it's okay. It's too small. It's an Octobedema. And then they inflate them all at once. And I believe they can have several kept in reserve there beneath their chin for when they need to replace their nets because they were clogged. Here's a rhizarian. Zoom in once on him, nice and close, please.
Go ahead. Forward. Zoom in. Okay, zoom in.
It's very transparent. So maybe it's not a 4am, maybe it's another rhizarian. We can try and get any closer in with a zoom to get more of an idea of its morphology, and then we can suck him up in one of the suction samplers. Nice and slowly, I think.
Yep. One in.
Move back, zoom out.
It's pretty close
Coming in
to us.
close,
Yeah.
yep. Be too close to suck up?
I don't know, follow him, feet. I'm slowly gonna rip. There you go. That's good. Suction on. Get suction? Yeah.
What type of rosaria is that?
That's a very good question. Not sure if I know what he is. He looks like the Hastagillirid, but he also looks different. There's a lot of diversity in rosarians that we're only just beginning to understand, and I wouldn't want to take a stab at such a complex group of organisms without really knowing. That's about right, I think. Too far in. No. Might have probably come all the way in here. Sure, we'll go find another one. Once they're that close, there's not much you can do. Oh, there's a Coelodendron. It's another rosarian there.
Are you good to keep going? I had the final watch, but if you're happy, it's fine. Go ahead. Oh, fish! You're missing the fish.
Missing the fish. Oh, we're squiddling over there.
I'm trying.
You're trying. Maybe I should be squiddling and you should be here.
Sure.
Yeah.
Creature. I catch that one.
So we're coming up at 2.
We're on deck at 2.
On deck at 2,
Yes.
and 3 more bottles.
Yeah, we need to do some rapid sampling of pretty much anything.
Okay.
The next 7 minutes.
In the next 7 minutes. All right. Everybody, keep your eyes open.
So how many do we have?
There's not many orchipluids
3.
around right now.
3 suction
6,
samplers.
7, 8. And the D samplers are all taken.
The no, D2 is open.
D2, we may have an orchipluid in it, but we're not
I
quite
think-
sure yet. But we should get what we can in the suction first.
Going.
There's the Coelodendron
Where?
in the sit cam. Here
That
comes.
here?
Oh, I'm supposed to be annotating that, aren't
Do we want to collect that? Just
Yes,
to,
sure,
Let's
we do.
collect that, please.
There's one
And
right there.
Alrighty.
Suction sampler, please.
Do we need any footage first?
Just
No.
enough of a zoom.
Yeah.
We've got one shot of it, I
I
guess.
feel like we've had so many.
Okay, that's good. Let's get in.
Now. This one's ready now. A little bit on. Sit. So. It's the brighter dot.
Okay. Push on forward and find another
Anybody
one.
see it? Yeah. Then, oh, there's a Solstice maybe? Oh. Yeah.
Sounds like a
Iraq.
Huh?
Keep.
Can always take silver.
Keep. Did we get it?
Good.
No.
Let's do some forward motion.
Rosarian. Push a little further
forward and try and find some big things.
Is there another one?
Oh yeah, another.
Oh yeah, let's start it, Andrew.
Sure.
Let's do it.
Let's just go for it.
Jenny, you don't have to be quiet. You're keeping us company.
On the over. If we had this in the arm, would have caught this right.
Little head paired. Do you guys want the DPIV on so you have another reference point?
And push that nozzle way, 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 way out. That should help.
That's right.
Push the nozzle forward.
You might be getting vehicle wash.
Yeah, I'm definitely getting vehicle wash.
No. Anybody still have eyes?
No, we're gonna have to
All
push
right, let's
forward, I
keep
think, see if we
searching.
can find another. Is that a Solmis? Is that a
Yeah,
Solmis or a Solmis?
something out here.
Forward.
A little faint.
See it?
No, I've lost it too.
Push forward.
Push forward.
Maybe we should like really race and see if we can find a large house.
Something.
Forward and up.
Oh, what do we have there?
Is that a fish up ahead to the left?
Something here.
About 2/3 of the way
Coming towards
to the left.
center.
Coming to the center now. There he is in the center. Push forward really fast now.
Where's that?
Speed forward, speed forward. He might be coming a little bit down. Speed
down.
forward,
Set.
a little bit down. Where'd he go? Where'd he go?
Something.
See it?
Lost it. What's in the center? House maybe? No. Yeah.
Still see it?
Yeah.
Nope.
No.
No, he was too fast. Why don't we try going fast like we were just then, though? See if we can find something big.
And forward. Forward and up.
Guess my fists. It's a Calicophrin. This must be Madagascar. A little bit further forward motion, please.
Roger that.
So then you'd stop.
Stop.
Can we stop on that and suck him in just as he is?
Yep. Camera.
Two of them.
Tighten up on the zoom a little bit.
Yeah, we got him. Okay.
I went in.
Good work.
Up.
Section sample of 6.
There it is.
There we go. One of them at least.
Yeah, they were clinging together, huh?
Yeah.
Yeah, I don't know where the other one is.
Thank you guys. Awesome.
There it is.
There we go. That's
Oh,
it.
great.
Suction off.
Wait,
That's
the
that.
two of them just came in. What came in before that?
Yes, we got 2 things.
Well, one
today.
of them had come in and then.
I think the first one might be a
Convection jaw.
platyskeleton hybrid amphipod.
Yeah, I think
Oh.
there's something else.
But
Bycatch.
the two-
We tried.
We got a bonus amphipod for the deal.
Index this out right to the next number.
Wait, do you see it coming down though? I haven't seen it.
Oh, not that one. Other way. Yeah.
I didn't see them come down. Hopefully they.
There?
So I'm afraid they might have gone up a bit.
Huh?
Can we do a small micro pulse now on 7 just to see if they're still left in the hose?
I think. Yeah, see, there's something there at the top.
Yeah.
Alright.
Suction's still on.
Yeah, it is, cause it's, I think it's stuck.
Is that tape again?
Yeah.
Maybe write a note on the sheet there
Turn on.
that
Yeah.
they might be in 6
Something's
or
off.
7.
I think that's.
I think that's them dangling into the chamber.
Should we advance to 8? Keep them in 7?
No, I think they're stuck
Oh, okay.
in the tubing. Or
When you take them out, just.
Move forward.
Push straight forward.
This is our time if you want to get on deck at 2. We gotta come up.
Okay. Let's be heading up. Heading up and forward.
You gotta recover if you want to get on deck at 2.
Yeah, Dougal, I think we have to start coming up.
Roger that.
If you give us just a little bit of forward when you're going up,
Move forward.
that'd be great.
Teeny forward.
Bridge ROV.
ROV Bridge, hello.
Hello, Tom. We are beginning our recovery.
Starting to recover, thank you.
Yeah.
Nice area.
Hmm.
Whoa.
Cool. That's so fun.
Screaming something forward.
Yeah.
What do we have? Oh,
Arsenic
sorry.
siphonophore.
Not totally salt chain, maybe.
Salt, okay.
That's what jumping.
Fish. Oh, there's another. What do we have up there?
See something? Coming up still.
Yeah. Oink.
30 meters. Nice area. Double espresso before processing those samples. You go 1:30 a.m. double espresso. Another rosarian. I'm surprised by how depauperate these fairly shallow, not surface. Nighttime, the surface should have a lot more going on, but it's like everybody's just left. It's like a ghost town as they've all gone and moved up.
It's too warm.
700 and 800, there were animals all over the place.
Google, Jenny in the chat wants to know how long does processing samples generally take?
Kind of depends on what it is and if it's known or new. That's
Big.
it's okay. It's not a giant squid.
What's that on the top?
That's a fish or a squid.
Fish? Can you take a little zoom in on that real quick? Just as we come up. It's very reflective.
Maybe a hatchet fish?
Yeah.
Yep.
A little hatchet. So bright.
He's not swimming up, he's like sitting horizontal, isn't he?
You want this?
Yeah, some
Should we
video would
try
be
and-
great. Whoa, 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 whoa. He's a bit
Oh.
He said, "No, you don't want this."
Shy. Keep him shy.
Coming up.
We're not
Yeah,
gonna catch
coming up.
him. So, undescribed species, because you're documenting everything about them, can take. Well, you've got to be quick, because they, while they're alive or recently dead, they self-digest and break down really rapidly. So you're working against the clock, so you can't take that long. But I guess 30 minutes or more per animal. The siphonophores, because there are so many zooids to photograph, are going to take an hour maybe each. I think we got 3 of them. And the colobanema, because we know what that is will take 10 minutes. So it really depends on what the animals themselves are, which is why we're making these decisions as we do the surveys. Do we really want to catch that this dive, or if it's common enough that we'll see it on another dive and we already have some difficult to process samples, maybe we'll leave it and catch it on a different day. Because we're all working on as a phallus callus and an apolemia, I think. We're all working on only a couple of hours sleep a night, so trying not to have too little sleep.
Right there.
The real challenge comes when the animal you need to document is too big to fit under any microscope or camera you have. I hear a laugh there because that's happened a few times. Or too small. Both are possible. Our colleague Manu has some great microscopes for photographing small things, so we're mostly okay for that. But we had some issues with equipment not arriving as it was supposed to, and we have a shortage of microscopes and cameras on the ship to be processing our samples. So several people on one microscope means it takes that much longer to process samples. Rosario. I guess we're doing a plankton net or a tucker trawl next, so everybody who's involved in processing those samples are now asleep, right? Or trying to sleep.
You should be if they're not.
Because they've only got like an hour or so before that goes in. Where that was. It's a bit like a Capitella.
Hmm.
We don't have Niskins this dive, do we?
No, no Niskins.
Agaripelicus. Nice. Go back and read it. Only got the family. I don't think you can spell that. Can you see? A-R-G?
ARGYRO
Ah.
P-E.
Oh, okay, found it.
Ah.
Agaripelicus. Nice one. Should be 50 meters. Horizon.
ROV great.
Horizon.
Go ahead, Tom. Hey Tyler, are you happy with me just staying where I am?
Yeah, there's hardly any current. Yeah, it looks like there's not much current, so if you feel anything weird, I'll give you a shout, but otherwise it should be okay as long as the lead's fine for the fellows.
Cool. If they want to lay, I'll give it to them closer to the time. Cheers.
Roger that, thank you.
Feet. Oh, something big there. Whoa, what was that? You really just-
That went to the bottom left corner?
Yeah, off the bottom left.
Hmm.
It's gone.
Yeah. Blackfish.
Yeah, I didn't really.
Does Fathomverse take into account neighboring frames in the same film? Like, well, I guess not the game taken into account, but like, let's say the model gets like a frame, and it's like, okay, this thing's probably this. On either side of that, there's gonna be frames of an object in the same area. But I'm just curious, like, does it take into account that, like, something that at one point in time was a really hazy little dot is now this thing?
It does.
That's really okay.
Yeah, so we track objects through time, and from those tracks we sample. We've changed this over time, but like we'll sample a few images along that track. And what we're trying to do is like present a number of different views,
Sure.
so that the models we train on diverse images of the same thing.
Yeah, yeah. Because I'm just thinking, like, all the times we spend searching, and we're like, oh, is it that? And we get there, and it's like, no, it's not that.
For sure. So we are doing that. It's just when we lose it from the field of view.
Yes.
We don't have anything special that is basically, stitching together those things.
Sure.
But if it's in the field of view, we're
Coming up
and so
300
some of the
meters.
things we would do is like, when is the object
From
the
this-
biggest?
Yeah.
That, select that frame grab, or, within, 10% of the start, or 10% of the end. So you want, obviously you want to be really careful because early on, it could be so small that you won't have any features.
Right, yeah, or it'll get lost in noise, or something could cross in front of it, and now you're tracking that new thing.
Yeah, if it's exactly, I mean, but it actually, I think that it's an object detector first, so we would lose an object if it intersected with another one, if they were the same thing.
Right.
But we would be able to recover
Yeah.
the information if they were different things.
Yeah.
But so that's why we have like an initial review of all the data. So like from hours and hours of video, we'll get, depending on the type of dive, anywhere from like 500 to 1,000 images to review, with these key features or key moments in time. And then because, we're working with TPLE or if we're hunting an animal in some cases, in most cases you'll get just like 3 to 4 images obtainable. But in these instances where we might be lose the animal from the field of view, that could increase a lot. And so we shut it down so much.
280 meters?
Yeah, I was just curious if they had a temporal aspect to that kind of track where they were prior to the main frame.
Yep.
Yeah, it definitely does. And one of the things we're trying to do too is track that temporal information, right? Because at the end of the day, we're going to want to be able to find out where in time. Sure. So, yeah, the content management system.
This one could probably, she's still watching.
Yeah. But we've tweaked it over time. I think we started out with, let's just see, 5 images or 2 images, just to, I think where we struggle with
Yeah, she's still
That'd be great, thanks.
Because if there are like 10 things in an image, you're gonna have 10 different tracks. And then those moments in time where it's the biggest will align, and so you can get many images of a really busy field. And so that takes a little more work for us to piece together.
I know in the game sometimes it's like
Hello.
2 things.
Yes, sir.
Hey, let me know when you're ready to transfer control.
About the other guy. So yeah, I imagine we can get very busy.
Up to you.
Thanks all for seeing the longer in case we see anything cool and all this. 50.
250.
Yeah.
Okay.
I have to say, the Midwater, these dives, it's been really quick for me to review, and the object detectors are working really well, even though it's mostly been trained. I think it's only been trained with very good at actually.
I mean, I think they're wrong to start, but you gotta do a little sign off or something, and then we'll kill them next once the clear term chatter starts. I mean, you can do it. Oh, well, I'm gonna be up until tomorrow.
250 meters or so. Like, All those surface squid have not kicked in yet, have they?
No, they're not.
197.
Okay, I needed to replace the battery. Okay. Okay. Waiting.
I'll go slow to stop and we'll hand it over to you at cool 260.
Okay. I'll take it over.
Alright, grab it.
Yeah, I got it.
Good.
Just 1 second.
Currents wise, wow. It's not much in weight currents. No, it's that's the least current in a while.
In good shape. Port arm shaded here.
Okay, ready.
Jeff?
I'm going to continue with the recover slowly.
Slowly.
Go.
Repositioning the ROV for recovery soon.
Yeah, that's port arm. Look on.
So, as you can see, the hangar is starting to get prepped for receiving the ROV back on board. We're going to be moving into recovery here and cutting the mics. Going to leave the cameras rolling until we're at surface, though, as per usual. And this is still not the last dive of this expedition. We got a few more days in us yet, so keep your eyes peeled for the next dive. Thank you for tuning in for this dive, and we'll see you next time.